Okay, we are starting now. Our first contestant for the day is Shruti S and S. Varshini. Give them a round of applause.
To explore this issue further, we have taken up a study on Kundangali.
Shape of BBMP. Also, over 1,000 tons of slag of waste.
To 2024 and observe the significant changes in the quality. Further, to check for the presence of microorganisms and chemical contaminants present in water and the system. We have collected around 4 samples from different points of the lake on different days and performed several on-site and off-site tests on it. The on-site tests performed include pH, temperature and turbidity. Whereas the off-site tests performed include alkalinity, hardness, chloride, and dissolved oxygen. Microorganisms is a polluted environment.
This shows the comparison of various off-site parameters which we have tested in a water sample, also affecting various organisms, especially species which are intolerant. Also, most of the water standards do not meet with those of BIS. Also, from our on-site visits, we could infer that the lake is extremely polluted due to human activity. Also, an unpleasant odor and green water could be observed due to excessive growth of
Released back into the atmosphere. This forms a continuous chain of events through which nitrogen is processed into many forms. However, a disruptance in this cycle may lead to eutrophication, which results in the water to be visibly green in color. This disruptance may be due to several reasons, including ammonia and phosphorus from the wastewater that is being released, and nitrogen from excessive rainwater. The simplest solution to this problem is that plants can absorb nitrate, which is a process form of ammonia, and create a balance in the ecosystem.
We would now like to recommend a few steps to improve the quality of water. Firstly, we could install pre-filtered drains which would help filter solid waste, then introduce beneficial bacteria which would help reduce the nutrient load, then add non-invasive native water plants which will absorb excessive nutrients, then keep the water circulated to provide oxygenation by installing fountains, water pumps, etc. These are few examples of biological filters, namely buffer zones, biofilters, floating treatment wetlands, constructed wetlands, as well as bioremediation, which help in reducing the nutrient load, prevent excess algal bloom, and help improve the overall health of the lake.
Floating treatment wetlands are small artificial platforms which allow aquatic plants to grow into them. These float on the surface of the water due to the entrapment of gas in rhizomes and interwoven roots, which can thus reduce the nutrient loads. These help by controlling the phosphoric as well as nitrogen content, as well as the microbes which are growing in this system form a biofilm or a sticky substance which helps in degrading the organic contaminants as well as absorbing the excessive nutrients. Constructed treatment wetlands is an efficient system of wastewater treatment where there is shallow ponds or channels which have aquatic plants that utilize physical, chemical, microbial and natural processes to treat the water. These help to create a control in nitrogen, phosphorus levels as well as the macrophytes that are being used, for example typha and phragmites, also help in absorbing the excessive nutrients.
In conclusion, there is severe degradation in both physical as well as chemical parameters of the water of the lake, urging us to take important steps to increase conservation of lakes by bringing awareness among people about the importance of lakes. Lakes are our breathing spaces, our lifelines. It is our duty to conserve, preserve, protect them. These are our references. We thank IISC for giving us such a wonderful opportunity, and our special thanks to our teachers, parents.
Thank you for the wonderful presentation. Can you come to your previous slides? Previous. Yeah, stop. What is the difference between Can you go back to that slide? Yeah. What are the identification here?
Kind of phase, solid phase and discharge.
Nice presentation. I just wanted to know what
Audience can ask if you have any questions. Next, Ganani.
Good morning everyone. My name is Maitri and I'm from Maharani Lakshmi Amani College. I live in Kodnathapura, which is very close to Bangalore. In that area, it is currently being developed and the farmland which is located in that area is on the brink of being urbanized but has not been urbanized yet and hence has a chance of being conserved. When I moved here 1 year ago, 3 months down the line, the surrounding area which once resembled a forest was slowly cut down, and instead of these trees, this land is being turned into new agriculture, into new residential plots as well as new roads. Farmland conservation refers to the practice of conserving agricultural land from being urbanized and using for non-agricultural purposes to conserve it for the future generations. Hence, this project is an attempt to try and conserve this farmland. I have two simple objectives. The first is to attempt to try and this is a comparison of before and after of how the surrounding area of the study area looks like. The yellow print is proving my objectives. The first is to collect samples. I collected 10 soil samples from various parts across the field and collected them in a small Ziploc bags. I then interviewed the owner of the farmland. I asked the farmer a few basic questions. The first was how much harvest he produced from this land. He said that he produced around 150 to 250 kgs of paddy every year from this land. I then asked him how much of this harvest he sold. So the farmer said that regardless of how much harvest he produced, 100 kgs of the harvest will be kept for the family as this is one of the primary courses of food for the family, and the rest they would sell for about 205 kgs. I then asked him if he grew any other crops in this area, as crop rotation helps in replenishing the nutrients in the soil. The farmer said that he did not grow any other crop except for ragi over here. Out of curiosity, I asked him why he did not. The farmer said that he did not have the necessary resources to grow any other crop here and has not done so for the past 5+ years. Since the farmer said that he did not have necessary resources to do a crop rotation, I searched for government schemes which could possibly help the farmer in acquiring more resources and increasing the production of the land. So the first scheme that I came across is PM Kisan Samman Nidhi. This policy aims to produce a direct income support of rupees 6,000 in 3 equal installments throughout the entire year. The second is PM Fasal Bima Yojana. This policy makes sure that in case of a natural calamity and the crops are damaged, the farmer is not forced to give up his land to debt traps or give it up for non-agricultural purposes. The third is a soil health card scheme. This policy helps the farmer in getting a free soil analysis done so that he can increase the soil efficiency and adequate. Here are the results of the soil moisture content test. As you can see, approximately 150 to 250 kg of ragi is produced from this land every year. The soil moisture content test shows us that the soil has a good moisture content capacity and also has a diverse soil diverse soil characteristics. The ideal pH range for ragi growth is 5 to 8.2. As the soil is of 5.02 as pH, we can conclude adequate and balanced. The organic carbon is 0.21. It is very low, but it can also be solved very easily by using manure, animal and cow dung, etc. In conclusion, urbanizing this farmland would be a loss for the ecological balance of the surroundings, as farmlands are one of the major resources of replenishing groundwater, which in turn helps the neighboring areas from water scarcity. And in return, this offers immense emotional value to the farmer and the family. So urbanizing this land would not be right. With this, I conclude my project. Here are the references that I used. In the end, I would like to thank IISC for organizing this event and giving me an opportunity to present this project. I would like to thank my institution, Lakshmi Amani College, my friends, and everybody who helped and supported me in completing this project. Thank you.
We have 1 more minute left. Any questions from audience? Yes, sir. Please. The first parameter pH. You have given the value 5.02. As far as the pH values are concerned, we know already from 0 to 14. But how can you get the fractional value, decimal value 0 to 5.02? And one more place also you have mentioned 8.2 something. How did you get the value? How can you calculate the value 5.02? If it is okay, 6, 7, 8, 9, 10, 11 like that. But how can you get 5.02 the value of pH?
So the sources that I used, they use decimal values to describe the exact pH value.
No, I am asking you, how did you calculate 5. To get the value 5.02, the decimal value?
Actually, I did the soil analysis at University of Agriculture Science, Bangalore. The results that I got from there is what I've put over here. I am not sure exactly how they concluded it in decimal values, but that's the result that I got from the soil analysis. So yeah.
Even up to degree level, we know already the whole value, that is 5, 6, 7, 8. I don't know how you got the value, that's what I was asking. Thank you. Reference. First one says NCERT Science Class 10. NCERT, what is that?
So in 10th standard, in the chapter acids, bases and salts, there's a method where you test the soil pH, and that's where that was my first step for doing the soil analysis for this project. And then in the very last evaluation of the round, I got to know that there's a very big soil analysis that needs to be done. So from 5 to 7.
What is the pH scale? Then approximately you can tell acidic and basic.
If I'm not wrong, it's from 5 to 8.
One more, you have mentioned only the ragi crop, right? So the area is 39,578 square feet area you have taken. And you have mentioned only 100 to 150 ragi, 8 kg. Then, okay. Please cross check the scale value for test. Okay, because they are work. Thank you.
Thank you.
Diksha, please come.
You can go.
Good morning to one and all present here. My name is Deeksha Liu from Mahalakshmi Amandi College, Malleshwaram. Today I am here to present my project on organic farming and sustainable agriculture at households. Introduction. Organic farming relies on natural methods without using chemical fertilizers or pesticides. And it could contribute to our healthier life if we grow our own vegetables at home. By growing our own nutritional vegetables and herbs, it could contribute to our healthier life and could reduce the expenses on groceries and objectives of my project are to utilize available resources efficiently and standardize the gardening techniques, to explore organic methods, for reducing pesticides and fertilizers, to study the needs and effects of organic farming if we do it at home. These are my study areas. My home terrace near Rajasinagar, Bangalore, and my grandmother's home terrace, Sanjeevani Layout, Bangalore. Essential materials were used such as pots, red soil, which was available near our house, cocoa beans, some seeds, and natural fertilizers were made in home with use of onion peels, banana peels, and tea powder, which is nitrogen, phosphorus, and potassium, which is very essential for plant growth. And natural pesticides were also using the mixture of onion, garlic, and chili powder, which all repels the insects and irritates due to the sulfur and antibacterial qualities of them. Methodology. The soil and components were mixed and put in the pots. All the seeds were first germinated and then planted in the soil. The natural fertilizers that were made in home were applied to the soil twice in a week with a ratio 1:10 in water, and pesticides were given to the plant regularly after the rain and for a week. All the plants were ensured to get sunlight for a year. Results. All the plants had got many leaves, rootlets, flowers, and many more. And we had even got harvest of amaranth leaves and radish leaves, which in current prices are 50 rupees in the market. We expect a 500 rupees save in every week by growing. These are my pictures of my organic farming. More nutrition can be achieved by using, and even the soil can sustain its fertility for a long time by planting legumes in the soil. Long term organic farming can be sustainable if we focus for future and grow and correctly plan what we should grow for, brings positivity in our mind and keeps our mind peaceful. Recommendation. I would recommend all the people in India and world to grow their own food in their houses, because it would not even disturb the commercial activity grown by the government, which even does not give loss to the government also. Practice how to grow and conclusion. I would like to say that by growing our own vegetables, we could harvest many fruits and vegetables which are healthier and more beneficial than those we brought from the store brought, and it could help.
Thank you. Any questions from audience?
Students can also ask.
Students, audience, all. nice. I think the healthier the soil, the better the plant grows. And also, neem, I think neem oil is also a good, one of the natural ways to use as a pesticide. So thank you, thank you.
The amendmented soil from various fruits.
Do you have home garden at your house? What do you grow?
I had grown in my home terrace using pots and
What did you grow?
in my home terrace using pots and
What did you grow? Which crop? What did you grow?
many leaves like orange.
Use this. Yes. So from this, what did you understand? Yes. So from this, what did you understand?
Sir, I understood that when we eat from vegetable or vegetables, the nutrients are less, and even after cooking, it will become more less nutrients. So
What do you mean by natural pesticides?
sir, which have mainly garlic and onion are naturally antibiotic and antifungal, and especially garlic contains sulfur, which repels insects and pesticides, and chili contains as it is irritating substance in it, it will other residues of present in our environment.
To what level, like how much percentage leadership is
It
possible?
depends on how many houses or how many people follow it. If more people follow, it will drastically reduce carbon footprints, but if few people follow, it varies from how
Any
many plants.
number can you give, like any percentage?
Plants are uncertainties. A very good morning to one and all present here. Today, IISC is about 19 acres. Methodology. The water samples have been collected from various points of the lake and have been tested in our college lab. The various parameters analyzed were pH, total dissolved solids, the is analyzed were pH, total dissolved solids, dissolved oxygen. For present chloride, then ammonium hydrogen, ammonium hydroxide, we get a clear solution indicating the presence of fluoride in the lake water. Presence of calcium, I've added sample. For a sample, I've added ammonium chloride, plus ammonium hydroxide, plus ammonium carbonate, which indicates by this base the same symbol indicating the presence of calcium in the lake sample. Presence of sulfate. For a sample, I've added barium chloride, plus excess of dilute hydrochloric acid. As a result, I got white precipitate indicating the presence of sulfate in the lake water. Confirmatory reduction. We need to reconstruct the natural water pathway to the untapped control structures of the lake. Measures to prevent soil erosion has to be taken place. Measures to prevent soil erosion has to be taken place. We need to clear the excess sediment of the soil as it can disrupt the wetlands function, due to which the wetlands volume will decrease. So regular cleaning of sediments, the wetlands volume will decrease. So regular cleaning of sediments is must. These sediments can be further used in many processes. That's from the local. So how they are involved in the maintenance of the lake. Lakes can be filled by vessels which act as a natural filter power which can be used as a renewable energy by the locals. Conclusion. From the above proposed solutions, it will be easier for us to conserve the lake Lakshmi Sagar. These are my references. Acknowledgement. My heartfelt thanks to the organizers and my dog.
So in the small lakes, do you think these are all possible?
Algal growth. If algae is grown, the fishes won't get oxygen. Dissolved oxygen content will be less. So this is for a bigger lake. So power generation for smaller lakes, it is not possible. So for bigger lakes, it's possible.
So you But is a bigger one or smaller one?
It's around 19 acres, sir. It's big. So at least 1 or 2 can do. But I didn't do the biological test for microorganisms and all. But plants are available around the lake.
Okay. What about fauna?
Good morning, one and all. Myself Nandini. Good morning, one and all. Myself Nandini. With my teammates, well-maintained soil enhances biodiversity by providing habitats for various well-maintained soil enhances biodiversity by providing habitats for various species and ecological balance. Wetlands around lake offer ecological services and environmental health. Maintenance of soil in this wetland area is essential for agricultural productivity, ecological balances, ensuring environmental soil sampling tools, example agar and soil. Sample containers, free site to soil analysis, pH meter, connectivity meter, soil test kits for nitrogen, phosphorus, potassium, etc. Notebook and pen, camera or smartphones for photographs, 2 mm mesh tape. Divide the lake into sampling zones based on inlet and outlet. Collect soil samples from the 3 points within each zone for this method we use to find the estimation of available nitrogen in soil. This method we use to find the estimation of available phosphorus in soil. This method we use to find the estimation of available potassium in soil. These are the expected results. This method we use to find the bulk density. These are the formulas. We work in lab. Soil sampling zones. Inlet 1, 7.6 pH, 0.28 EC, 135 TDS, 27.7 temperature. Sampling zones. Inlet 1, 40% sand, 33.33% clay, 26.6 20.43% clay, 27.15% silt. These are the glaser analysis method. pH levels range 6.8 to 7.6 indicates slightly acidic to neutral soil. Favorable for most plants. Electrical conductivity EC range 0.28 to 0.53 microsiemens per centimeters reflects moderate soil concentration within the acceptable range for plant growth. Total dissolved solids TDS range 135 to 211 mg per liter, slightly elevated levels possible due to proximity to the lake and mineral accumulation. Nitrogen, high levels at inlet 1 and outlet 1, 201 to 300 kg per acre. Lower levels at inlet 2, 50 kg per acre, indicate potential nitrogen deficiency. Phosphorus, low levels across all sites, particularly at inlet 2, 1 to 3 kg per acre, and outlet 1, 25 to 49 kg per acre, suggesting a possible limitation for plant growth. Potassium, higher at inlet 1, 11 to 15 kg per acre. The lowest at outlet 1, 4.7 kg per acre.
indicating variations in nutrient availability. Inlet 1 has the most sand, which means it drains water very well. Its soil is loose, making it easier for plant roots to grow. Outlet 1 has the most clay and silt, which helps the soil hold water and minerals. The soil is more compact, which might make it harder for roots to grow. Inlet 2 is in the middle with balanced amount of sand, clay and silt. And soil is moderately compact, offering a good mix for plant to grow. These are the awareness gallery. The soil health around Bilwakali Lake is integral to maintaining the wetland ecosystem, which provides the soil of Bilwakali Lake has a neutral to slightly alkaline pH 7.5 to 7.6, which supports nutrient availability and healthy plant growth. Low salinity is indicated by EC values 0.28 to 0.78 microsiemens per centimeter. We extend our heartfelt gratitude to our headmistress for the opportunity, our guide teacher for their guidance, Dr. Vinay sir and Vigilance sir for valuable support to this project, EWRG CES IISC Bangalore for their advice.
This one. Sand, clay, silt. How do you differentiate and what does this value says? Sand, clay and silt. How do you differentiate sand, clay and silt? So what is sand, what is clay, what is silt?
See, when water flows, it forms a layers, right? Different layers, right? So looking into the point of that, we can also differentiate sand, clay and silt. Even you can tell in what way you understood. Even looking at the sizes or looking at the way it slippery way. Okay, that's it.
Sir, ಅಲ್ಲಿ condition ಆಗಿರ್ತಾರೆ. Sir, ಅದಕ್ಕೆ ಅಲ್ಲಿ ಸ್ವಲ್ಪ clay ಆಗಿರುತ್ತೆ.
Okay.
ಅಲ್ಲಿ ಜಾಸ್ತಿ ಖನಿಜಗಳು ಎಲ್ಲ ಇರೋದ್ರಿಂದ
ಅಲ್ಲ ಕ್ಲೇ ಅಂದ್ರೆ ಕ್ಲೇ ಯಾವ ಟೈಪ್ ಇರುತ್ತೆ? ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಬಿಟ್ವೀನ್ ಸ್ಯಾಂಡ್ ಅಂಡ್ ಕ್ಲೇ ಗೊತ್ತಾಯ್ತಾ?
clay ಅಂದ್ರೆ ಸ್ವಲ್ಪ ಮುಖಕ್ಕೆ ಒಂಥರ
ಸ್ಟಿಕ್ಕಿ ಟೈಪ್ ಇರ್ತದೆ, ರೈಟ್? ಸಾಫ್ಟ್ ಇನ್ ನೇಚರ್, ರೈಟ್? What is the pressure it is when you compare to clay?
Clay, silt ಅಂದ್ರೆ
Okay, no problem. So we have done wonderful work because lot of data is there. But again, observations are also very important. You have to observe the things. Thank you.
otherwise
Excuse me, there is one question for the moderator.
Which method you use to differentiate? Use seal and which had highest value? Do you have any photos of that? And which had highest size, particle size? Size wise, which was highest? Which sand, clay or seal? That is why okay. That's what he asked actually. What is the difference? The size wise, sand have the highest, clay if it's diameter wise, it is smaller. Okay, they are asked in different ways, same.
Good work, lots of data. Much over. Okay, moving on to the next project.
Next, MD Sohil and group. There is a pointer also, you can use that if you want to highlight it. Please start.
Good morning, Honorable President here. Myself, Abhishek from class 10, with my partners Vignesh and Pavitras. We are from Vidyanjali School. I thank Honorable President to allowing us to present our study, analysis of oxygen production in leaf. Plucking a single leaf from the plants of the roadside may seem insignificant, but it impacts on the environment more than we realize. Plants plays a crucial role in oxygen production through photosynthesis by absorbing carbon dioxide and releasing oxygen. We rely on for survival. Removing even one leaf from a roadside plant, it slightly reduces the plant's ability to carry out this vital process and leading to the less oxygen production. Beyond the oxygen production, plants also support biodiversity, help purify water and act as a buffers against environmental hazards.
To calculate the oxygen production of individual leaf from various species of plants, educate the community on how small action like non-plucking leaves can support a healthier environment. Our study area is Vipas Colony located Bangalore 56008. The materials which are used in our project are healthy plant with multiple proteins, graph sheets, calculator, mobile phone for taking pictures, notebook and pen for recording data. Calculation of the average oxygen production per leaf based on individual measurements. To check total leaves grown and the height of the plant in our study period. Reference ISO Nature Model Karnataka State Council for Science and Technology 2016 IISC.
This is the plant used in our studies. Neem plant, West Jasmine, Common Zuva, Cluster Pig, Ogamia pinnata, Rose plant and Hibiscus. This is the method that we have calculated oxygen. This is the checklist of our study. The common name of cluster fig was Indian fig tree and its scientific name was Trochilus. Area of a leaf in square centimeter was 85. Amount of oxygen per liter per year was liter per year was 260.61. Leaves grown in study period was from June 6th to July 28th. The common row of plants saw an increase of only 12 leaves over nearly 2 months, 28 to 40 days. The rose plant grew significantly from 47 leaves on June 6th to 107 by July 28th. But this growth took time. Leaf plucking is a quick action, but leaf regrowth will happen slowly. Indiscriminate plucking disrupts the natural, gradual growth process of the plant. Patience and care will needed to ensure the plant growth thrive without unnecessary stress. Let us discuss about our results. This study shows human well-being. Neem and hibiscus with the moderate oxygen per annual. With this flowers beauty added to the society.
These are our awareness gallery. The awareness campaign highlights that small action like plucking a single leaf from the roadside plant can reduce the plant's ability to produce oxygen. By preserving roadside plants and their leaves, we can help support a healthier environment, particularly in urban areas with limited greenery. Community members must understand the cumulative effect of such action lead to decrease the oxygen production
and negatively impact overall air quality. This campaign encourages to protect all forms of vegetation and to be mindful of their impact on urban green space.
Reinforce the message that action like plucking of leaves from the plants may produce a less decrease in the oxygen production, whereas especially in urban areas, whereas greenery is limited. By this, avoiding unnecessary plucking of leaves can make a significant difference in human well-being and healthier environment. Selecting and planting the right species in urban and rural areas can maximize the oxygen production and we can get environmental sustainability and human health. We thank our HM and guide teacher for allowing us to conduct this study, and we also thank to Anushri AS for this framework, and Guru Prasad KL as a group member of this study, and we also thank for Sri Vijula sir for guiding us, and we also thank for
Wikipedia.org. ISO Nature Model Karnataka State Council for Science and Technology 2016 IISC. Nature Science Internship Program Manual 2017-2021 Adamya Chetana. Tyson and Zygat 2010 Plant Physiology 5th ed Senior Associates. Thank you.
Species like.
Sir, we buy small saplings from the shops and we planted in our house and we observed them and we were taking pictures of daily whether it's growing or it's stopped.
Okay, all the plants, previous slides you have shown as a how they grow. Go back to that slide. Yeah, next, previous. These are the plants, right?
Yes, sir. Only leaves, sir.
Only leaf.
Yes, sir, only leaves.
Because I believe your words. Go back again, observe that diagram. Yeah, I discussed because I have seen all guava, everything, even most of the plants. If you see the leaves, then come back to the statistics values, right? Yeah, you have mentioned 50 centimeter height. Huh? No, we are talking about leaves, right? You are talking about that is the leaf size.
Area of leaf is
Water diagram ಒಳಗಡೆ ಬಾಕ್ಸಸ್ ಎಲ್ಲ ಕೌಂಟ್ ಮಾಡಿದಾಗ ನಿಮಗೆ ಏರಿಯಾ ಆಫ್ ಲೀಫ್ ಸ್ಪೇಸ್
50 centimeter you are looking there.
ಸರ್ ಐದು ಸರ್
area of a leaf is 12 centimeter.
Square centimeter means what? Square centimeter. Leaves, when you go back to the data, maximum you will find 5 centimeter. Would find 3, remaining 3, right? The rows.
ಸರ್ ನಾವು ಅರ್ಧ ಬಾಕ್ಸ್ ನ ಕೌಂಟ್ ಮಾಡೋದು ಸರ್ ಆಫ್ ಜಾಸ್ತಿ ಇದ್ದಾಗ
But still, when you say, okay, square area, what about length you told? Height. Here you tell that box, height discuss, height how much? But that matters because statistically when you are comparing the values, okay, you have to be careful. Leaves and plants, right? Okay. Not taken. Thank you.
Anybody has a doubt?
So as somebody who doesn't know about this. You said that on an average 65 liters of oxygen is lost every year if I pluck one leaf. What does that actually mean? How much oxygen is consumed by a human in a day?
I cannot tell perfectly how much human need for a day, but human needs 7 trees oxygen for survival.
In a day?
In a day. I don't know. I didn't study. We are in study. We didn't complete a 8-hour study.
In general, which tree gives more oxygen?
Let's moving on to the next project. Lavanya IIS. After that, Satvika Insarati, please be ready.
A very good morning to everyone present here. My name is Lavanya from Maharani Lakshmi Academy College. So, let me introduce you to my topic. Soil has enormous life forms and dynamic habitats. Soil provides shelter for some vertebrates, microorganisms like bacteria and fungi. Soil supports our ecosystem objectives to explore different species in selected fields to field study, to understand the interactions and roles of microorganisms in soil, to understand the role of microorganisms in soil as fertilizers, the importance of living microorganisms in soil. This is my study area. Materials and chemicals used. Methods. Cook 12 test tubes, 12 micro pipettes and petri plates, 2 to 3 vessels. And serial dilution technique. Serial dilution technique is done to dilute the concentrated soil solution. 4-plate technique, incubator and stay room temperature of 25 degrees Celsius to 27 degrees Celsius for 3 days. And organisms was also observed in soil. Results. This is commonly known as earthworm. They are classified as decomposers and they cannot survive in harsh and hostile conditions. Night crickets. Usually crickets come under nocturnal insects. They are known for their loud chirping and they hide in warm places during the day. And this is earth snake. This is its common name. Its scientific name is Indotapus braminus. It's a blind snake because its eyes are 2 dots, which is covered with translucent and hairy and antique legs. They chase down prey rather than catching on the web. Dung beetle. They are fascinating creatures that play important role in breaking down dung and they feed on feces. Beetle help in dispersing. This is results for fungi and total number of fungi was of 4 types and the most abundantly present fungi was Fusarium. Results. This is result for bacteria is gram-positive or gram-negative. Discussion. Organisms and microorganisms help in decomposition of waste in soil. They help in exchange of gases like oxygen and nitrogen. It helps plants to get all possible nutrients. Microorganisms act as fertilizers by providing primary nutrients to host plants. Documentation and survey was started in the month of August 12 and to September 27. Time of survey was between stop cutting trees and waste of chemicals. Dung beetle help in disposing of seeds.
Dispersal of seeds. How?
Yeah, because they usually feed on pieces which contain seeds, and it doesn't mean that they eat it up. They leave the residue items also.
So, म्हणजे हे next आणि हिमानी
A very good morning to one and all present here. A very good morning to one and all present here. My name is Anand. Building relationship and bonding between humans, with nature, and divine, nurtured by many past scientific roots, linking us to the past, present, and future with terms of religious culture, ethnic heritage, and ecosystem. India is a home to many sacred roots, lands and lungs are present. In India, 40,000 in India, 1,424 in Karnataka, and 2,000 approximately are present and identified and studied so far. We have to understand sacred root is a case-sensitive chain reaction of practical difficulties protecting, managing, and conserving issues. What is anti-compromise?
Something is not there. Okay.
So coming to the objectives, our main objectives was to study the physical changes and emerging trends present in the study area. And the second objective was to explore the role of sacred roots, from the ecological and the social perspective. The Nagabana or the snake god group, which is totally concretized. This is the satellite or the drone view. Coming to the Chamundibana or the deity group. First of all, the Chamundibana or the deity group is totally natural and is not affected by any human activities. And the name of the deity worship here is the Chamundi Deva and the area. Coming to the threat information, there is a lot of habitat loss and water scarcity has been observed in this particular study area. And one of the main reasons, Nagabana or the snake group, the name of the deity worship is Naga god, and the extent is 0.004 hectares. And first of all, we would like to bring a comparison here between the earlier state and present state. Earlier, you could observe that it was completely natural and it was worshipped in the foot of the trees. But right now, it has been commercialized and concretized. And this is the concrete structure that you can observe here. This is the study site. Coming to the research methodology, we conducted personal and community interactions for our first-hand information. And we also
15 participants whose age ranged from 11 to 87, and there were 5 females and 10 males that we interviewed. And data analysis was grounded on a variety of parameters such as hydrology and topology. And we identified 5 main stakeholders. They were the custodians, priests, localites, and the youth. These were the questions that we interviewed and asked the people. Coming to the study observation, the climate that we observed in this particular area was of a tropical monsoon, high rainfall, and hot summer. And coming to the hydrological aspect, the Chamandibana basically it has a water body which is 150 feet in diameter and 6 feet in depth. And coming to the Nagabana, it has a nearby stream which is now dried up because of the construction of a dam. And coming to the Madaka, the most important part, it is a traditional water harvesting system, which in which the water, these are the herbs, climbers, and this is floristic diversity. And we also observe different species of fungi. And this is the list of the endangered and threatened species according to the IISC and Red List. Coming to the research findings, we observed that coming to the recommendations, we would like to recommend some measures for the further protection and considering it as a heritage site is very important. And next, exemption of land tax and in this project, we have explored the cultural heritage related to the sacred groves, and we would also mention that the sacred groves are a divine home that are meant to be revered and respected, and so we would like to promote further preservation and conservation of sacred groves. These are the references. Acknowledge the IISC team and our youth teachers and black organization. And we would also like to suggest a special thanks to Mr. Udai Kumar Shetty of the Caligula Biological Park for helping us in the identification of medicinal plants. Thank you.
Please have the butterfly slide.
So which are butterflies and which are moths in this?
So the moth is Apis atlas. It is widely observed in the Western Ghats and it's very common. And the other ones are all butterflies.
Butterflies and moths. That is a different about it. Previous slide. Did you capture these photos?
Yes, all the photos are captured by us, none of it is internet by.
Using which camera?
We use a phone camera.
The phone cameras, you got this.
Yes, you can see the watermark.
Not on this Kingfisher one.
Yeah. Actually, it was also captured by my cousin's photography camera.
And that is not a swallow bird, the first one. Please check twice. You have to put wrong species name. Either you have to change the image or a name. It's a black brown bird. It's not a swallow bird. Okay. And previous one also not this great blue heron. Please do check twice. And that is also not a forest owl. Forest owlets are very small. That is huge. Please do check. Next slide. Here. Reptiles, snakes and lizards and amphibians. What do you call all these reptiles and amphibians both put together? I add peptides and amphibians together. That's what.
Next, Maharshi and team.
And today we are going to welcome topic study of freshwater algae. Introduction about algae. Algae is a group of diverse organisms that can be found in oceans, lakes, puddles, waterfalls, trunk of trees, swimmers, etc. They play a major role in producing oxygen in the environment. They also play a very important role in photosynthesis. They are also used to lake. The latitude and longitude are as shown. Second sample was collected in Bangalore Palace. It was near Bangalore. The visited time was 11:34 a.m. and the visited day was 8 September. The third sample was collected in Bhagat Park. A small portion of algae was placed on the slide. Dissecting needle was used to investigate of the lake. 2 to 3 drops of crystal results of our study. The first sample, which is located near Dwarkapuri Hall, we found Pseudophiliaster boreanum. It is a eukaryotic species and its characteristic converts into oxygen and it removes hexavalent and chromium from contaminated aquatic ecosystems. This is a clear image of it. The second species which we found in the same sample was Diatoms, which is eukaryotic species and its particular species. So diatom is earth, the substance composed of fossil diatoms. It is used as filters in genetic veins and in the veins in diatoms in dynamites, recurring regions, and it also has antibacterial and anti-cancer compounds. They have various industrial and pharmaceutical applications. This is a clear image. And the third, location is Common Park. We found one species, Prospera, which is a prokaryotic algae and is a freshwater species. It is used as a nutritional supplements to aquatic organisms due to its rich compounds in sugars and root systems. And in agriculture, it plays an important role when they are used as fertilizers and soil stabilizers. Example is cyanobacteria. And it can remove heavy metals and organic pollutants. Example is Pseudophiliaster boreanum. And in biofuels, since that is an attractive source of lipids, proteins, etc., it is used in biofuels. And in food and agriculture, microalgae have nutrient compounds like vitamins, proteins, fatty acids, etc., which are used as nutrients. Example, urea. So the conclusion which we draw from this study is microalgae cultivation offers sustainable solution for biomass production, pollution control, and economic growth, positioning it as a key factor for industrial and environmental advancements. Further research of large-scale algae should be advanced, and this approach can not only support environmental sustainability but also contribute to the economic growth as they are used in biofuels, animal feed, and other algae-based products. Acknowledgement, references, to advanced research, what we told in the conclusion. And anyway, we are already focusing on cleaner solutions for fuels, so it will be a very good addition.
The answer is already there in your discussion.
Yes.
Can you see that it contains the carbohydrates. Carbohydrates, these are the major fraction. If you want to produce biohydrogen, you require sugar fraction.
Scientific name is it diatoms?
We have not identified the particular species, so we
No.
found.
Is it diatoms I'm asking? Like
Yes,
scientific
yes.
name. Scientific name is it diatoms?
No, it's a common name.
Then why you put it under scientific name? It's a common name, okay? Under this many species are there. Cosinotis, Navicular, etc. So you should be very specific about that. And you know common name of cyanobacteria?
Cyanobacteria is a common. So blue green algae.
Blue green. Like you mentioned it as a fertilizer.
Yeah.
So there are many like cases have been like identified that cyanobacteria causes blue and it is very toxic. So you can also mention under this contaminated list also.
is study of targets called? Psychology.
Good, good. Next is Varshita MS and Meenu M, Flora O, Amani Campus.
Good morning to one and all. I am Varshita and myself from Maharani Lakshmi Amani College.
So our presentation today is based on flora of American campus. So our main agenda of today is to give introduction, methodologies, what were our objectives, thesis and our conclusions.
Firstly, introduction. Floral diversity. Floral diversity.
Then to classify them on basis on exotic, indigenous, ornamental, medicinal or growth and to know the trend of plant growing the college.
Our college measures an area of 3,635.29 meters square and a perimeter of 261.11 meters.
Methodology. So we had done a field study on plans of our college campus.
Spider plants, rose and tail flower.
For example,
Sprain's feather, which are used for treatment of ulcers and tuberculosis, also the treatment of stomach ulcer.
PCDC, which is responsible for treatment of bee stings.
African tulip tree, which is used for epilepsy and convulsion control.
Then comes hibiscus, which helps in reducing your hypertension and helps in
These are indigenous. So the difference between exotic and indigenous plants are the plants which are not native to a specific region, but this can survive and
conclusion. By the help of our study, we are not only able to find good traits of the plant, but also some bad, such as spartanium, which was some as a contaminant from the United States of America, which is infecting us genetic risk or genetic diversity, which can refer to variations which occurs in the species. Due to this, there is a loss of genetic diversity. Maybe our chain of evolution may disturb in this. So what we would like to conclude is, let each plant has its own uniqueness. So we would like to allow all the plants to grow by its own. People must be aware of the importance of plants which are grown widely, and we should promote to grow them.
Thank you for the presentation. Can you please come?
actually before we could find out the rest of them, we couldn't. We lacked focus.
You should be very careful with your answers because these are scientific forum. And also you told that content plans.
So for aesthetic purposes and just for decorations, we use for further.
Okay. Next, come to your previous model slides. Audience, any questions? We'll break for tea. It is 11:15. By 11:30 we'll be back. 15 minutes will be enough, I guess. Okay. Tea is served and
Okay, we will start the session again. Vijeta, Shreya, please come.
And I'm Vijeta, and we're from Maharani that listen to things like global warming, air pollution, lung problems, and who's the main cause of all of this? Carbon dioxide. Just like how you see kids getting attracted to every single new toy you find in the stores, the same way industrialists are attracted to new industrialists and they spend their money on factories, and most of these factories mainly emit a large amount of carbon dioxide. So here that before industrialization, it is completely less. And in the beginning of the industrialization, it is completely less. And in the beginning of the industrialization around 1900 to 1950, it had a little increase, but it was still 1900 to 1950. It had a little increase, but it was still sustainable. But after that, it kept increasing, and it still is. And this is India's carbon dioxide emission. 90 it had almost a quarter of it, and reaching more than 20 billion. And now on an average every year, there's about 35 billion tons every single year. And it had become a little less during the COVID period, but again it has increased.
Manipal Naturals was a study area where we found a place where CO2 is emitted. Vacuum tray dryer.
The supporting file was also sent.
Taking the carbon dioxide out of the industries. So now we're going to take the carbon dioxide from the chimneys that's going to come from the industry, and we're going to use a chemical solvent, which is a porous solid material. For example, a fluid gas, and it's going to be passed through an amine solvent, and it binds the carbon dioxide so that it's separated from the other gas. So we have pure carbon dioxide. Now this gas supplied to other places, and when it comes back to its own form, we can see that there's one extra product that is carbonate. So that's where it's going to come from, and here it's going to get purified, and we're going to have pure carbon dioxide, and this is going to be the cathode, anode, and the electricity, and it's going to come back. So this is the equations, and it's going to be repurposed in the industry, so we're going to have it's more economic. And then other benefits of this is there's going to be a huge amount of greenhouse gas reduction. And then the lithium batteries are usually flammable, but this is not flammable. So it's going to be more secure. So when we see about a lot of 48,000, so we can conclude that about 70 to 80% of the carbon dioxide are growing to become more developed. We are also conserving the carbon dioxide and going to more ecological side of the world. And this reuse of carbon dioxide was achieved by using the fuel that's going to be used in the batteries. These are the references we've used.
Thank you for the presentation. What is your innovation in this?
The innovation is that this was mainly, it's not yet come into use, but a main company had got up this idea. So we found out that this carbonate that is extra, rather than throwing it, we can implement it, and it's going to attract more people to use this methodology. And we want to tell a lot of people about this because not a lot of people know about this technology. So we want to make people aware about this. So that's what the main goal is.
If you want to create awareness, what awareness program have you conducted?
I just have a question. In this individual case study, like all of these are profit driven at the end of the day, so they only invest in things like this to help them in the long run. So how much would it cost, a rough estimate of cost to build a system like this for the case study that you did?
Around $1,000, but then it's the profit what will attract their industrialists, and they would want to use it more.
ಅಂಶಗಳ ಪ್ರಭಾವ, ಆಹಾರದಲ್ಲಿ ರಾಸಾಯನಿಕಗಳು. ನಾವು ಪ್ರತಿದಿನ ಸೇವಿಸುವ ಕೃತಕ ತ್ವರಿತ ಆಹಾರದ ಬಗ್ಗೆ ನಿಮಗೆಲ್ಲ ತಿಳಿದಿದೆ. ತ್ವರಿತ ಆಹಾರ ಅಂದರೆ ಬಿಸ್ಕೆಟ್, ಚಿಪ್ಸ್ ಅಂಡ್ ಪ್ಯಾಕ್ ಮಾಡಿದ ಆಹಾರಗಳನ್ನು ತ್ವರಿತ ಆಹಾರ ಎನ್ನುತ್ತೇವೆ. ಅದರಲ್ಲಿ ಸುಮಾರು 80ರಷ್ಟು ರಾಸಾಯನಿಕ ಪದಾರ್ಥಗಳಿದ್ದು ಅದು ನಮ್ಮ ದೇಹಕ್ಕೆ ಸೇರಿದಾಗ ಹಲವು ಸಮಸ್ಯೆಗಳನ್ನು ಉಂಟು ಮಾಡುತ್ತದೆ. ರಸಾಯನಶಾಸ್ತ್ರವು ನಮ್ಮ ಪ್ರತಿನಿತ್ಯದ ಜೀವನದಲ್ಲಿ ಹಲವು ಕೊಡುಗೆಗಳನ್ನು ನೀಡಿದ್ದು ಅದರಲ್ಲಿ ನಾವು ಪ್ರತಿದಿನ ಸೇವಿಸುವ ಆಹಾರವು ಪ್ರಮುಖವಾಗಿದೆ. ನಾವು ತ್ವರಿತ ಆಹಾರದ ಬಗ್ಗೆ ಚರ್ಚಿಸಬೇಕಾದರೆ ಅದರಲ್ಲಿ ಸುಮಾರು ಲೆಕ್ಕಕ್ಕೆ ಇರುವಷ್ಟು ತ್ವರಿತ ಆಹಾರಗಳಿದ್ದು ನಮಗೆ ಎಲ್ಲ ಆಹಾರಗಳನ್ನು ಒಂದೇ ರೀತಿಯಲ್ಲಿ ಒಂದೇ ಸಮಯದಲ್ಲಿ ನೋಡಲಾಗುವುದಿಲ್ಲ. ಅದಕ್ಕೆ ನಾವು ಗೂಗಲ್ ಸರ್ವೆ ಮಾಡಿದ್ದೆವು. ಅದರಲ್ಲಿ 3 ಪ್ರಶ್ನೆಗಳನ್ನು ಕೇಳಿದ್ದೆವು. ಅದರಲ್ಲಿ ನೀವು ಮೊದಲನೇದಾಗಿ ಚಾಟ್ಸ್ ಅನ್ನು ಸೇವಿಸುತ್ತೀರಾ ಎಂದು. ಅದರಲ್ಲಿ ಸುಮಾರು 96.4% ಜನ ಹೌದು ಎಂದು ಹೇಳಿದರು. ಆಮೇಲೆ ಯಾವ ಬಗೆಯ ಚಾಟ್ಸ್ ಅನ್ನು ನೀವು ಹೆಚ್ಚುವರಿ ಸೇವಿಸುತ್ತೀರಿ ಅಂತ ಕೇಳಿದಾಗ ಪಾನಿಪುರಿ, ಮಸಾಲೆ ಪುರಿ ಅಂಡ್ ಸಮೋಸ ಅದು ಮುಖ್ಯವಾಗಿ ಕಂಡು ಬಂದ ಜಾಸ್ತಿ ಸೇವಿಸುವಂತಹ ಚಾಟ್ಸ್. ಹಾಗೆ ನಾವು 3ನೇ ಪ್ರಶ್ನೆಯನ್ನು ಕೇಳಿದ್ದು, ನೀವು ದಿನನಿತ್ಯದಲ್ಲಿ ಸೇವಿಸುತ್ತೀರಾ, ವಾರದಲ್ಲಿ ಎಷ್ಟು ಬಾರಿ ಸೇವಿಸುತ್ತೀರಾ ಹಾಗೂ ತಿಂಗಳಿನಲ್ಲಿ ಎಷ್ಟು ಬಾರಿ ಸೇವಿಸುತ್ತೀರಾ ಎಂದು. ಅದರಲ್ಲಿ ನಮಗೆ ಹೆಚ್ಚುವರಿ ಕಂಡು ಬಂದಿದ್ದು ವಾರದಲ್ಲಿ ಹೆಚ್ಚಾಗಿ ಸೇವಿಸುತ್ತಾರೆ. ಉದ್ದೇಶಗಳು: ಪಾನಿಪುರಿ ಹಾಗೂ ಮಸಾಲೆ ಪುರಿ ಸೇವಿಸುವುದರಿಂದ ಆಗುವ ದುಷ್ಪರಿಣಾಮಗಳು ಹಾಗೂ ಅದರಲ್ಲಿ ಬೆರೆಸುವ ರಾಸಾಯನಿಕ ಕಲಬೆರಕೆಗಳು. ಪಾನಿಪುರಿ ಇದು ಅತ್ಯಂತ ಪ್ರಿಯಕರ ಆಹಾರವಾಗಿದ್ದು ಇದನ್ನು ಎಲ್ಲರೂ ಸೇವಿಸುತ್ತಾರೆ. ಪಾನಿಪುರಿ ತಿನ್ನುವುದರಿಂದ ಅತಿಸಾರ, ನಿರ್ಜಲೀಕರಣ, ವಾಂತಿ, ಬೇದಿ, ಹುಣ್ಣು, ಜೀರ್ಣಾಂಗ ವ್ಯವಸ್ಥೆಯಲ್ಲಿ ಅಡಚಣೆ ಹಾಗೂ ತೀವ್ರವಾದ ನೋವು, ಕರುಳಿನ ಉರಿಯಂತಹ ಸಮಸ್ಯೆಗಳು ಉಂಟಾಗುತ್ತವೆ. ಮತ್ತೊಂದೆಡೆ ಪಾನಿಪುರಿಯಲ್ಲಿ ಹೆಚ್ಚಿನ ಉಪ್ಪನ್ನು ಬೆರೆಸುವುದರಿಂದ ಇದು ನಮ್ಮ ರಕ್ತದ ಒತ್ತಡದ ಸಮಸ್ಯೆಯನ್ನು ಹೆಚ್ಚು ಮಾಡುತ್ತದೆ. ನಾವು ವಿವಿಧ ಪಾನೀಯಗಳ pH ಪರೀಕ್ಷೆಯನ್ನು ಮಾಡಿದೆವು. 3 ಬಗೆಯ ಪಾನೀಯಗಳನ್ನು ನಾವು ತೆಗೆದುಕೊಂಡಿದ್ದೆವು. ಮೊದಲನೇದು ರಸ್ತೆ ಬೀದಿಯಲ್ಲಿ ಮಾಡುವ ಪಾನಿ, ಹೋಟೆಲ್ಗಳಲ್ಲಿ ಮಾಡುವ ಪಾನಿ ಹಾಗೂ ಮನೆಯಲ್ಲಿ ಮಾಡುವ ಪಾನಿ. ಅದರ ಬಣ್ಣಗಳ ಹೆಸರು: ರಸ್ತೆ ಬೀದಿಯಲ್ಲಿ ಮಾಡುವ ಪಾನಿ ತುಂಬಾ ಹಾನಿಕಾರಕವಾಗಿದ್ದು ಅದು ಹೆಚ್ಚು ಕೆಂಪು ಬಣ್ಣವನ್ನು ಹೊಂದಿತ್ತು. ಆಮೇಲೆ ಹೋಟೆಲ್ಗಳಲ್ಲಿ ಮಾಡುವ ಪಾನಿ ಸ್ವಲ್ಪ ನೀರಿನ ಪ್ರವಚನ ಇದ್ದಿದ್ದು ಅದು ತೆರವಾದ ಕೆಂಪನ್ನು ಹೊಂದಿದ್ದು. ಮನೆಯಲ್ಲಿ ಮಾಡುವ ಪಾನಿ ಕೇಸರಿಯಾಗಿತ್ತು. ಅದರ pH ಸಂಖ್ಯೆ ಪಾನಿಪುರಿಯಲ್ಲಿ ಮುಖ್ಯವಾಗಿ 3 ಬಗೆಯ ರಾಸಾಯನಿಕಗಳನ್ನು ಬೆರೆಸುತ್ತಾರೆ. ಅವು 3 ರಾಸಾಯನಿಕಗಳು ಸನ್ಸೆಟ್ ಎಲ್ಲೋ, ಬ್ರಿಲಿಯಂಟ್ ಬ್ಲೂ ಹಾಗೂ ಟಾಟಾ. ಮೊದಲನೇದಾಗಿ ಸನ್ಸೆಟ್ ಎಲ್ಲೋ ಕೇವಲ ಪಾನಿಪುರಿಯಲ್ಲಿ ಮಾತ್ರ ಅಲ್ಲ ಹಾಗೂ ಹಲವು ಬಗೆಯ ತ್ವರಿತ ಆಹಾರಗಳಲ್ಲಿ ಅದನ್ನು ಬಳಸುತ್ತಾರೆ. ಅದನ್ನು ಜಾಮ್ಗಳಲ್ಲಿ, ಚಿಪ್ಸ್, ಬಿಸ್ಕೆಟ್, ಐಸ್ ಕ್ರೀಮ್, ಜ್ಯೂಸ್, ನೂಡಲ್ಸ್ ಹಾಗೂ ಪಾನಿಪುರಿಯಲ್ಲಿ ಬಳಸುತ್ತಾರೆ. ಅದರಿಂದ ನಮಗೆ DNA ಹಾನಿ, ಹೈಪರ್, ಹೊಟ್ಟೆಯಲ್ಲಿ ನೋವು, ತಲೆನೋವು ಹಾಗೂ ಮುಂತಾದ ಕಾಯಿಲೆಗಳು ಆಗುತ್ತವೆ. ಬ್ರಿಲಿಯಂಟ್ ಬ್ಲೂ ಅದನ್ನು ಕೇಕ್, ಮಿಠಾಯಿ, ಚೀಯಿಂಗ್ ಗಮ್, ಡೈರಿ ಉತ್ಪನ್ನಗಳು ಹಾಗೂ ಪನ್ನೀರ್ ಮತ್ತು ಪಾನಿಪುರಿಯಲ್ಲಿ ಅದನ್ನು ಬಳಸುತ್ತಾರೆ. ಅದರ ಪರಿಣಾಮಗಳು: ಕುರುಡತನ, ಅದು ನಮ್ಮ ರಕ್ತ ಸಂಚಾರದಲ್ಲಿ ಸೇರುತ್ತದೆ, ಚರ್ಮದ ಕಾಯಿಲೆ, ಕಣ್ಣು ಮಂಜು ಮುಂತಾದ ಕಾಯಿಲೆಗಳು ಬ್ರಿಲಿಯಂಟ್ ಬ್ಲೂ ಅಂತ ಅದನ್ನು ಮಿಠಾಯಿಗಳು, ಹಲವು ಬಗೆಯ ಜ್ಯೂಸ್ಗಳಲ್ಲಿ ಹಾಗೂ ಐಸ್ ಕ್ರೀಮ್ ಮತ್ತು ಪಾನಿಪುರಿಯಲ್ಲಿ ಟಾಟಾ ಅನ್ನು ರಾಸಾಯನಿಕ ಬಳಸುತ್ತಾರೆ. ಮಸಾಲೆ ಪುರಿಯನ್ನು ಸಾಮಾನ್ಯವಾಗಿ ಪಾನಿಪುರಿಯಿಂದ ತಯಾರಿಸುತ್ತಾರೆ. ತಲೆನೋವು ಹೆಚ್ಚಾಗುತ್ತದೆ. ಅದರಲ್ಲಿ ಬಳಸುವ ತರಕಾರಿಗಳು ಶುದ್ಧವಾಗಿರುವುದಿಲ್ಲ. ಆದ್ದರಿಂದ ನಮಗೆ ಹೊಟ್ಟೆಯಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳು ಸೇರುತ್ತವೆ. ಸಮೋಸ ತಯಾರಿಸುವುದು ಮುಖ್ಯವಾಗಿ ಆಲೂಗಡ್ಡೆ ಬೇಸ್. ಅದರಲ್ಲಿ ವಿಷಕಾರಿ ರಾಸಾಯನಿಕಗಳಿವೆ.
ಹೈಡ್ರೋಕಾರ್ಬನ್ ಮತ್ತು ಕ್ಲೋರಿನ್ ಗ್ಯಾಸ್ ಪರಾಕ್ಸೈಡ್ ಅಥವಾ ಇತರ ಬ್ಲೀಚ್ಗಳಂತಹ ರಾಸಾಯನಿಕಗಳೊಂದಿಗೆ ಸಂಗ್ರಹಿಸಿದ ಮೈದಾನಿಕನ್ನು ಈ ಸಮಸ್ಯೆ ಮಾಡಲು ಬಳಸುತ್ತಾರೆ. ಎಣ್ಣೆಯನ್ನು ಹೆಚ್ಚಿನ ತಾಪಮಾನದಲ್ಲಿ ಕಾಯಿಸಿದಾಗ ಕ್ಯಾನ್ಸರ್ಗೆ ಕಾರಣವಾಗುತ್ತದೆ. ಪದೇ ಪದೇ ಇಂತಹ ಆಹಾರವನ್ನು ಸೇವಿಸುವುದರ ಮೂಲಕ ನಾವು ಮಧುಮೇಹ, ಅಧಿಕ ರಕ್ತದೊತ್ತಡ, ಹೃದಯ ರಕ್ತನಾಳದ ಸಮಸ್ಯೆಗಳಿಗೆ ಮತ್ತು ತೂಕ ಹೆಚ್ಚಾಗುವಂತಹ ಸಮಸ್ಯೆಗಳಿಗೆ ಗುರಿಯಾಗುತ್ತೇವೆ. ರಾಸಾಯನಿಕಗಳು ಅಜಿನೋಮೋಟೊ ಅಥವಾ ಇದನ್ನು ಮೊನೋಸೋಡಿಯಂ ಗ್ಲುಟಮೇಟ್ ಟೇಸ್ಟಿಂಗ್ ಪೌಡರ್ ಅಂತ ಅಂತೀವಿ. ಆಹಾರದ ರುಚಿಯನ್ನು ಹೆಚ್ಚುತ್ತದೆ. ಸಾರಾಂಶ, ಆರೋಗ್ಯವೇ ಭಾಗ್ಯ ಎಂದು ನಾವು ಹೇಳುತ್ತೇವೆ. ಆರೋಗ್ಯಕರ ನಮ್ಮ ವ್ಯಕ್ತಿತ್ವದ ಮೇಲೆಯೂ ಪರಿಣಾಮ ಬೀರುತ್ತದೆ ಎಂದು ಸೈಕಾಲಜಿ ಹೇಳುತ್ತದೆ. ಪೌಷ್ಟಿಕಾಂಶಗಳ ಕೂಡಿದ ರುಚಿಕರ ಹಾಗೂ ನಿಯಮಿತ ಆಹಾರ ಸೇವನೆಯು ಅತ್ಯಗತ್ಯ. ನಾವು
ಓಕೆ, ಸೊ ಪಾನಿಪುರಿ, ಬೇಲ್ಪುರಿ, ಸಮೋಸ ಎಲ್ಲ ತಿನ್ಬಾರ್ದು ಅಂತ ಹೇಳ್ತಿದ್ದೀರ. ಬಟ್ ಇಮೇಜಸ್ ಎಲ್ಲ ಯೂಸ್ ಮಾಡಿದ್ರಲ್ವಾ, ಕೆಲವೊಂದೆಲ್ಲ ರೆಫರೆನ್ಸ್ ಆಫ್ ಡೈರೆಕ್ಟ್ ಇಂಟರ್ನೆಟ್ ಇಂದ ತಗೊಂಡಿರೋದಿದೆ, ಅದಾದಷ್ಟು ಅವಾಯ್ಡ್ ಮಾಡಿ ನೆಕ್ಸ್ಟ್ ಟೈಮ್. ಎನಿ ಟೇಸ್ಟಿ ಕ್ವಶನ್ಸ್ ಫ್ರಮ್ ಆಡಿಯನ್ಸ್?
I don't the same college, but what I would like to tell the audience is that add the fruits, add the grains, add the grains, add the all the sprouts. It's the most healthiest and the most inviting option for the kids. And you can also use even potato. What she was talking about the potato is I agree with her. Because I'm a North Indian and I use lot of charts, and that's one best thing. The potato where the roots start growing, that is the most unhealthy one. You should not be using that. The fresh potato, when it's boiled and used immediately with all the other fruits and the sprouts, nothing like it. Please go home and try it. You'll have it.
So you had mentioned about the chemicals which were used in the past.
I get that information from a research paper.
What are the toxins present in ayurveda? Because you have mentioned the poison is actually it is not poison.
ಸೊ ನೀವು ಪ್ರೆಸೆಂಟ್ ಮಾಡಿದ್ದು ಹೊಡೆಯೋದು ಆದರೆ ಕೆಲವೊಂದು ಮಾಹಿತಿಗಳು ಇನ್ನೂ ಬರಬೇಕಾಗಿತ್ತು ಅನಿಸುತ್ತೆ. ಅದನ್ನು ನೀವು ಇನ್ಕಾರ್ಪೊರೇಟ್ ಮಾಡ್ಕೊಳ್ಳಿ ತುಂಬ ಮುಂದಿನ ದಿನಗಳಲ್ಲಿ.
ನೆಕ್ಸ್ಟ್ ಖುಷಿ ಭಾರ್ಗವ.
Good morning to all the judges and audience. I am Khushi Bhargava from IISC National Public School, and I am going to be presenting my study on ants as bioindicators and ecosystem services. So ants are eco-friendly, eusocial insects of the family Formicidae and belong to the order Hymenoptera. More than 13,800 species in the entire world have been classified till now, out of which I will be analyzing 8 species in this presentation. Also, ants serve many ecological roles to the environment, and I will be mainly focusing on ants as bioindicators in this presentation. My main objective is to analyze patterns of ant species richness and their assemblage structure, and then examine the utility of ants as bioindicators of future climate change. So there are two factors which I'll be
altitudinal patterns and then their response to sulfur dioxide levels. And also I would like to experimentally determine a possibility of changes in the chemical nature of ant species due to pollution in the environment, which I'll be talking about later. So overall, these are the methods which I used for my study. First, I collected the samples by hand picking or leaf litter collection, and then I took pictures of my collections. Then I identified the species by noting down their physical structure, and then in the later part, I checked their pH value. This was my study area, a small part of my school campus, and the pointers show which ant I noted in which area. So this is the first analysis data which was collected in my school. Over here we can see that the most abundance of ants is of basement ants, and it was found near the branches of an infected gooseberry plant. And then all of this data was found in whatever study area I have shown. Now this is my analysis data too. It is a part under the IISC project which was done in Queensland, Australia, from 300 to 1100 meters above sea level. This approximately same collection methods were used in this paper as well. This table shows some of the common species which even I noticed in my study area and which were listed in the paper which I found from Queensland. This graph shows that the abundance of ants is more when the altitude is less. At 300 meters above sea level, we see that the common black ants, Camponotus complexus, is maximum in abundance, and Peperamonium caesitum is the least. Yeah. There are two conclusions which can be drawn from this paper. The first is that assemblies of most ants reduces with an increase in the altitudinal level. And second, that with an increasing level of sulfur dioxide, the ants number decreases. This is the next analysis data, which was in Jammu Kashmir, near the Himalaya region. This is a graph that shows the abundance percentages of different ant families in this area. This table shows how many specific numbers of ants were noted in that area. So we can see that this species, it is not found in the abundance of 10,000 meters, 1,000 meters above sea level, but it is found in a greater abundance at 2,000 meters above sea level. And that's why we can classify it as a very high altitude ant. And the others also, we can see that this one, which is specific to the Himalayan region, is more at the 1,000 meters above sea level region. This graph shows the same data. Now, the conclusions drawn from this analysis is that same as the altitude increases, the abundance of ant decreases. Now we will get back to the data which I collected in my school. So we get the 10,000 meters above sea level area. So over here, we can note that in the first species over here, the school, in my school, the abundance was more as compared to Australia or Jammu and Kashmir. One reason for this can be the temperature, and assemblies also reduced with low temperature. So because Jammu and Kashmir and Australia are colder places than Bangalore, so that's why we can see that there are lesser ants in those areas as compared to my schools. Now, leaving the altitude factor aside, I also wanted to check how the surrounding components can affect the assemblies of ants. So I conducted a small experiment to check in different body parts. So I use a litmus paper and another method. In the litmus paper, I slightly brush the ant and then I observe the color changes on the litmus paper in the head region and the abdomen region. In the head region, I notice the correct color, which is a pink color for indicating the acid. But in the abdomen region, I notice a blue color, which tells that there is a base present. So then I concluded after repeating the experiment that these were the changes which I noticed, that there might be some mechanism in the body of the ant which causes this change. Like from acid in the head, it changes to base in the abdomen. Or it can be even affected by the gases in that area or the ant's diet, which causes a basic color in the abdomen. These were some additional observations which were related to my study area. And this is an experiment which I did in the preliminary stages of my experimentation. They show only the reaction of abundance of ants in two acids, oleic acid and vinegar. Now to conclude this project, I would like to suggest some things. We see that the abundance of ant species changes with the external conditions, and so it is a very good plan to use these results to classify ants as bioindicators. So for this, we need our scientists to work more in this field and then come up with specific patterns seen in the environment of different regions. And then what we can do is, because ant species abundance decreases with growing altitude, we can use these in different areas to maybe predict some natural disasters in the which might occur in the future. We might predict these, and so this would help in reducing the cost of several measurement instruments if we find specific patterns to map this. The overall conclusion is that ants have clearly the ability to reflect on ecological change and their abundance and density changes in different regions. They can also be used as ecosystem health indicators after a substantial research, as I told. These were the references I used to make this presentation. I would like to thank each and every member who was directly or indirectly supporting me through this presentation. Thank you.
Thank you for the presentation. What is study of ants known as? Study of ants. Mammatology is right. So you have chose your school campus and directly flew to Australia, then Himalayas. So how do you correlate this different terrain, different?
What I wanted to do was at the same altitudinal level, and I wanted to take different areas with some thought that there might be something related to the chemical nature which is changing after sulfur dioxide enters their water. So that's why I wanted to test that what is actually happening to the experiments.
So there is some pH changes and that what you observe, finding. So there is a change in pH.
Yeah. So what I found out was that in the head, I could find basically it is indicating it might be because of its diet. Because of its diet.
Right. So there is no SO2 influence in that.
Till now I could not find it, but I'm working on it.
Next we have Tejas and team. Tejas, Vihan, Muthukumar, Atal. Next, Arun, please be ready. Nagapan Arun.
Jai Shri Gurudev. Good afternoon, esteemed judges, teachers and participants. I, Tejas, along with my teammate Prihan of Class 10H, stand before you to present our topic, documenting and evaluating biomass contribution of overseas species. Urban vegetation includes trees, plants and shrubs that are grown in parks, streets and backyards, which play an important role in mitigating the carbon emissions as well as regulating the microclimate. The World Health Organization recommends about 9.5 meters square of green space per person. And to counterbalance the per capita CO2 respiratory rate, there's a requirement of a minimum to 32 to 55 trees per person. With this in our mind, our project highlights the role of trees in managing and reducing the carbon emissions. Our objectives include making up an inventory of tree species and to measure and compare the biomass of the abundant tree species and to evaluate their role in carbon sequestration through our biomass calculation. The methodologies include mapping the tree study areas, data collection where we collected the tree diversity in the tree study areas and presented in the form of graphs, evaluating the biomass of the five most abundant tree species and classification of trees. Further, we have given few recommendations, that is the methods for enhancing green cover.
So for our project, we have selected three areas of study. The first study area is in Shantiketen layout in Alkere. The second in Meenakshi layout in Banarghetta main road. And third is in our school, BJ Sasha Public School in Bhima. So the following slide shows the data illustrating the population of plants and their species in a graphical format. This is for study area 1. So in study area 1, in 4 distinct parts in study area 1, we have employed the quadrat method to calculate the plant population density. This method involves dividing the study area into equal size quadrats and averaging the number of plants, average the population of plants over these areas, which results in the plant population density. This data we collected in study area 2 and in study area 3. Then we found that in Shantiketen layout there are total of 352 trees and 36 species. In Meenakshi layout we have found 499 trees with 17 species, and in our school we have found 177 trees with 27 species.
Coming to the biomass calculation. For the biomass calculation, one must require the basic physical attributes of the tree, being the height of the tree in feet, for which we use a DIY clinometer, and the girth of the tree, which must be measured at a height of 1.37 meter above ground. Step 1 includes calculating the above ground biomass, where you use the given formula. Step 2 includes calculating the below ground biomass, which accounts for about 20% of the above ground biomass. Hence, the total biomass comes up to 1.2 the above ground biomass. Step 4 includes calculating the total dry weight of the tree, which accounts for about 72.5% of the total biomass of the tree. Step 5 includes calculating total carbon weight, which is the weight of the carbon dioxide, which accounts for about 50% of the total dry weight. Step 6 includes calculating total carbon sequestered of the tree, and hence the ratio of carbon dioxide to carbon, which is 3.667, is multiplied the total dry weight of the tree, and we get the total carbon sequestered by each tree. We have used the given formula to calculate the total carbon sequestered in the three different areas. In study area 1, the top 5 tree species by number as follows, and the badam tree was able to sequester the most amount of carbon, being 1216.50. Similarly, in study area 2, the Hongemara was able to sequester the most amount of carbon, which is 853.97, and in study area 3, it was the mango tree, which was able to sequester about 128.78 kgs.
The importance of tree biomass. Tree biomass is basically the amount of living material contained in a tree, including its trees, including its trunk, branches, leaves, and roots, usually measured as dry weight. Some notable ecological importances are like the carbon sequestration, that the trees have carbon helping to reduce atmospheric carbon dioxide and helping to combat climate change, and use as an renewable energy source. The tree biomass can be used, reducing the reliance of fossil fuels and help in water irrigation. The importance of prominent tree species. In the year 1973, there were 979 tree species recorded in Bengaluru, and in the year 2014, by a study conducted by IISC, it found that there are 117 distinct tree species. And in our study, we found that there are 43 distinct tree species. The following table shows some notable ecological importances of the top 5 tree species, like the African tulip tree, also African purity, it acts as a pollinator powerhouse and it stabilizes the soil. And the Ashoka tree, it acts as a natural air purifier. The Badam tree, it helps supports health and supports digestion. And the Honge Mara, it's a drought resistant crop and helps to improve the biodiversity of the region. And Nilgiri tree is known for its anti-inflammatory properties.
Now, after understanding the importance of tree and tree biomass, a question arises: how many trees are actually enough? To know this, one must know the per capita CO2 respiratory emission ranges from 192 to 320 kg per year. Knowing that 1 hectare of forest sequesters about 6 tons of carbon yearly, this means that one tree on an average is able to sequester about 6 kg of carbon. Hence, to offset 192 to 320 kg of CO2 per year, there is a requirement of at least 32 to 55 trees per person. But this is not able to be we're not able to meet this required standard due to the urbanization that is taking place. We have calculated number of trees per person study area 1, which is approximately 3 trees per person, which is a clear example of urbanization that is taking place and the number of trees are decreasing.
So in this in study area 1 in Karthikeyan layout, we have found that there are 499 trees and 17 distinct species. And in Meenakshi, and in study area 1, there are a total of 352 trees with 30 distinct species. The possible explanation for the higher number of trees in area 2 is due to the presence of lakes in close vicinity. And in our school, there are 177 trees with 27 distinct species. Among the tree study areas, we have noticed that Hongema glabra, also known as the Hongema, was the most abundant tree species. And then all the trees we have found, we have classified them into avenue trees like the Badam and the Ashoka tree, windbreakers like the Ashoka tree and sacred fig, medicinal plants like Moringa and Nilgiri, road dividers like the Neem tree and sacred fig, and sound barriers like the bamboo people. In conclusion, from all of these study areas, 15 more abundant tree species sequester around 34,610 kilograms of carbon per year, offsetting the carbon emissions of a typical urban petrol car, which emits about 2,000 to 3,000 kilograms of carbon dioxide per year, and a diesel car emitting about 3,000 to 4,000 carbon dioxide per year, highlighting the importance of expanding green spaces and reducing urban air pollution.
Recommendations. We would encourage the colony associations to tag trees with the vernacular as well as scientific name, as well as their role that is their ecological significance to the biomass calculation we have done. Additionally, we would suggest them to undertake a plantation drive for planting native tree species. We have undertaken an awareness program under which the tank pump is study area 1. We have collected the different tree species and presented in form of a QR code, which when scanned will produce a document which one can use. This is a gallery, that is a biomass calculation that we have done in our school. References, the notable ones are the IISC report of 2014 that we have used and the Google Earth that we have used for spatial analysis. Thank you.
Thank you for the presentation. So what is your research in this project? What is your research in this project? Trees are there, they are documented. Yes. What research did you do in this project?
So research basically through our curriculum that we have been learning, we just understood that trees are important for us, but not many people have researched on which ways. So we have undertaken that, we have researched that carbon sequestration is one of the main channels through which it's able to, it has an ecological importance to that manner. So that is, doing that research we have taken this way.
Okay, thank you. Any questions from audience?
You mentioned about bamboo tree. So what is your view on the importance of bamboo in carbon sequestration? So is you know what is the total forest cover in India that is covered by the bamboo forests? So is there any significance in since you already focus the bamboo and the people, these two trees focus on that. So what is your view on that?
Not sure about carbon sequestration, but as a sound barrier, it's able to capture more higher frequencies and hence it's able to help in that manner. But we'll get back to you on that, sir.
Audience.
Thank you. There's a small announcement. Please do not leave because we have certificate distribution. Already your certificates are ready for your presentation. And if your friends have gone to other hall, please ask them to come and assemble here. So who are presented here, we'll have a certificate distribution after all presentations. Thank you.
Next, respected moderators and your audience. Must shine in three sustainable development goals of the United Nations, 7, 11 and 13. These mainly deal sustainable development goals stand. So now I'm going to be explaining to you guys where which is something that we can capitalize on as a developing nation. Now, you can see this is India's generation chart over the past decade. In 2009 and 2010, we had a generation of 805 billion units, whereas in rural areas or urban areas, meaning they don't consume more electricity. So India's generation demand is always going to grow. There's always going to be demand and consume more electricity. So India's generation demand is always going to grow. There's always going to be demand, and it is up to us to meet that demand. If we meet that demand in a sustainable fashion, it will be helpful for us. The amount of energy that India needs, the amount of energy we are generating is not matched by renewable energy. The amount of energy that India needs, the amount of energy we are generating is not matched by renewable energy generation. We are again, just always been in an energy deficit. In the financial year 2009-2010, the deficit was negative 10%. In financial year 2021-22, the latest data that the government is published, it is negative 0.4, meaning that we are still in a deficit. Government is published, it is negative 0.4, meaning that we are still in a deficit. So we need to move faster and step up and over 50% of our renewable energy needs is solar energy, and we are aiming to reach 2,80,000 megawatt hours solar energy, and we are aiming to reach 2,80,000 megawatt hours of solar energy generation capacity by 2030. It's increased by 30 times in the last 9 years. This is all done through the National Solar Mission, which is a policy of the Union government. Now, another major thing I'd like to talk about is biofuel-based energy, something that is overlooked in today's world, which is something that can heavily contribute to our fight against climate change and our fight to switch towards renewable energy. Now, it's not always going to be sunny. It's not always going to be, for example, there in Cato where it's sunny, whereas Leh Ladakh is not sunny. So we need to be able to transmit this energy. We need long-range transmission cables. This also needs high level investment. There's a lot of market opportunity, and there's inconsistent policies. When governments change, the renewable energy policy changes, and everything goes for a toss. For India's transition in COP26, under the pledge that we gave, we probably mentioned the National Policy on Biofuels 2018 amendment in 2022, the National Solar Mission Union Government project. The renewable process obligation basically means that DISCOMs, that is the distribution companies, have to purchase a set amount of renewable energy in the energy that they are distributing, and net metering, as the people of solar plants at home, solar panels at home, they can sell energy back to the grid. Now, there is youth perspective. This is a survey that I did, and there is youth support. Over 70% of youth believe that it is a high priority, so there is a youth support. Now, I would like to do my very own part and carry out an analysis at our school level, and I found out that our school spends approximately 2 lakhs per month on electricity bills, and we use 26 megawatt hours of electricity. Now, if you analyze the roof they cover the area, so we actually set up solar panels on the roof, we should be able to generate 611 megawatt hours. So this means that we are generating double of our electricity we can sell back to the grid and use the money to develop the school maybe or in the supply, and large energy storage systems like flow batteries. These are my references.
Nice presentation. So you are diving into the renewable energy sector. It's very nice to see. So you gave the two renewable perspective. One is solar, another is biofuels. In terms of transportation, okay. So there were solar cars and also even now they are moving towards the biofuels. So what do you think which will be more feasible? The solar or we move towards the biofuels?
Sir, I personally think solar will be a little more feasible from a market perspective, from an economic perspective.
The Toyota, Tata
Yes,
Motors
sir,
also
they
coming
came
with
out
that.
with that, yes.
So the flex fuel vehicles are already there. And even Karnataka is also no, they are also been utilizing the biofuel, biodiesel blending.
Yes, and they set up a new policy that came out about 3 months ago, the state government policy.
Nice, thanks.
What about discharge of solar panels?
The European Union already started investing in disposal mechanisms. We haven't. So we can either buy the technology from there. Thank you, sir.
So good morning to one and all present here. I am Shaurya of 10th standard with my friend nicknamed as Garbage City because garbage is found everywhere and BBMP has literally failed to manage the waste that is being produced everywhere and BBMP has literally failed to manage the waste that is being produced. So what can we do for it? We all love people when we think that the waste produced in our house is our responsibility and not the civic authorities. A rapid increase in the consumption and production processes societies generate as well as reject solid materials agriculturally, industrially, institutionally. These wastes are called as solid waste. Solid wastes are usually the main objective of a study is to understand the amount of waste generated. So we selected our neighbors, Sheila Auntie's house and asked her to store 1 week's waste. The materials used was a weighing scale. After a week, we went there and collected the waste and weighed it. It weighed 750 grams. We were surprised. Absolutely not necessary. Our school is in the BPMP limit and every day when we all leave our houses for work or at school. So hence all the waste in another houses also will go to the roadside. The following alternative methods were adopted by our Auntie to reduce the amount of dry waste generated in our house. So instead of shampoo sachets, she started buying shampoo bottles so that bottles can be recycled. Same case, give it to the vendors who would come near her house. Instead of that, she could also get some vessels. What we found out was that in Google, we would get about 4,000 to 25,000 per hair, like per kg of hair. But the vendors who came near our houses would only give about 100 rupees for 100 grams of waste hair. And one more thing, let me make clear here that we also found an empty wheat flour cover. And normally wheat flours in India, they are added with potassium bromate. Let me tell you that it's a carcinogen as a preservative to increase the shelf life of the flour. So carcinogen refers to the chemicals or the preservatives that are cancer causing. So Sheila Auntie understood this and started to buy whole wheat. So I recommend all of you who all are here to also use whole wheat instead of packets. And we also asked her to store plastic bottles and juice bottles so that it could be stored and sold to these scrap dealers. And at the last visit, we only got 180 grams of waste. We found kebab masala packets and etc. masala packets and of course food containers. What she said that was, she didn't know to make the masalas and she purchased it. But I failed in convincing her to stop buying food from online. What we found out was that out of 750 grams of waste, 300 grams of waste was of online food, which is absolutely not necessary. Do you think that our grandparents used to buy from online like Zomato or Swiggy like us? Nowadays, as per TVRAMACHANDra said, and request her to reduce the amount of plastic generated in her house. And we also gave her 5 cloth bags. We used to go to floor mill instead of her. And we also convinced her to go to grocery shops where they used to pack groceries in paper covers. Hence, I recommend all of you to avoid online purchase of food and
only an awareness. He could have been moved one more step, how to use these wastages to convert useful products, right?
Sir, this was about disposal of the waste, which I had concern of.
Okay.
So, like we have to dispose the waste in the right.
Segregation of wastages, and based on segregation, we can able to reduce the wastages in future, right?
Yes.
Then, see, highly impossible to, 100% reduce the usage of plastics in the country.
It's not possible, sir. Even in my house, I only generate 100 bottle, you can refill it or keep some plants in that, and that can be recycled.
So most of the cities now, municipality people are doing that one, segregating the things, and plastic they are sending to the plastic industries.
Thank you. Any questions from audience?
Why does government, vendors come near rural areas who collect it and they give it to keratin extract factories and like that, wig making. So they give it there and make their fund.
Okay. And one more question. You somehow convince that auntie of yours.
Yeah.
But then how are you going to convince the entire country of it?
Actually, this was just this thing, awareness about that, how I reduce. So I will recommend this, like it is possible if you can. You have to change your mindset before you do it. So you have to have that mindset they have to do it. So that's what I am here for. The conference.
See, it is everybody's responsibility. It is everybody's responsibility, and we can't change the mindset of any other people. So it is everybody's responsibility. We have to start with our own. Like he started with convincing one person.
That's what you
So,
are
yeah.
A mindset of some people who I would recommend to say that their mindset would dominate in their household also decrease like that. I was not focusing only on rural areas.
Okay, so
I respect
even so many scientists are from rural areas.
Yeah, I respect rural areas. That is.
New startups, they are coming with that, and they will be very happy to see as a young are coming with such ideas, and you can provide them. Actually, they are correct, like this is also one of the source, so it will be very
Which can be further recycled.
Supermarket, you need to buy it from a packet itself.
No, like let's say buy in a larger quantity. So you get boxes. Like
Even
using
if they get in boxes, even if they get it.
Okay, this is a suggestion.
Okay,
One more suggestion, I asked you a question, no, that how are you going to convince the entire country? There is one solution which I also propose somewhere else. So you will make a device in which whatever kind of waste you are getting is segregated.
There was a party from other venue. If any participants are there who is present in other venue, they are asked to go there.
Only
Presently, my name is Fauzia. I'm a student of class 10 standard from KT English High School, which has an area of 33.9 acres. It has a mixed landscape. At two places there is water here too. Me and my friend visit this place every Sunday and take care of plants. The main objective of our study was to understand the habitat of birds and also the impact of habitat loss. The material used was Sony Alpha and Canon, the two cameras, and also the mobile. This picture shows the planting of saplings in the year 2019. A part of the lake by 10,000 square feet is allotted for a school to develop as butterfly park. This year, on Environment Day, we started planting saplings. In reality, we noticed so much of chirping of birds. It sounded like. With the help of my teacher, I identified it as biodiverse senior Mr. Chandra Prakash. He made us understand more about those beautiful birds. And we also we listed 23 different types of birds that by a river, robin, sunbird, white fan, and etc. This is the Baya River bird's nest. There are many interesting facts about Baya River. First and foremost, the big township nest which will have an entrance at tubular end. This nest are woven with long strips of paddy leaves. And we have also male bird build a nest. Female bird comes for inspection. If she likes, she will enter inside and the male will complete the nest. If she rejects, then he has to build new one. In fact, I also try to make a nest by using a dry leaves between nest. All things are done with the managed. Loss of water bodies will certainly lead to loss of all these bird species. We are lucky to see them now in their habitat, but our grandchildren will have to see them only in bird sanctuaries. But and human activities should be completely prohibited in the areas like
it should be done. It should be done.
Recently, I saw on one high tension wire a pigeon sat, and on that spot only, it died. So how can these birds
male only will complete this. Female will accept and give. Female will accept. He will only will. If she receives, then he will get another one.
Then he has to try. But in case of bird, once it is accepted by a female, both male and female will work together to complete the nest.
Okay.
And last you came to your electric towers. How come it is a difference in same study area? Last how is the 19th slide and 21st slide. Up little, go to previous slides. Here. Where was this photo captured?
Nakumli.
Okay. Next, come to our electric towers. Next slide. Next.
This photo we got.
This was just a message.
Yes.
You should be very specific.
Okay.
Thank you. Session you please leave. Probably after this presentation, we'll have lunch break. Later, you can continue. Okay? Yeah, it's over to you now. Spurti and Lekitati.
lost. We learn about environmental awareness in years, but everything is ending up in books or a topic of a symposium or a session. It should not end up like that. But in case we can do it like good, educational institutions can get lot together, and we wanted to show the commitment of ours towards nature, mother nature, what we have done in our campus to make it green. We wanted to spread environmental awareness, we wanted to make the waste eco-friendly, we wanted to build accountability for each and every person, each and every action. We wanted to minimize the waste generation, we wanted to conserve the energy, we wanted to share our ideas which we have done in our campus, our campus. There are several activities which we have done in our campus, but there are the best 10 of them we have offered to share with you guys. And the first one is water conservation. You have added aerators to the taps to check the outflow of water. We constantly interact and insist the small children in school so that we can minimize the water waste. And we promptly report if there is any leakage in campus that to staff, and staff get it checked as soon as possible. And our school has a inverted check pipe with holes in the cellar garden. Whenever the aquarium water gets cleaned, once in 15 days, that pipe which is used to remove the aquarium water is drawn into this inverted shaped pipe. Through the holes, the cellar garden is leaked. And we have one more wash, and we have one more L-shaped pipe in other side of the garden, which is directly connected to the wash basin. And we have also rainwater harvesting system in our school. We have kept the old school vehicle tires to give our garden an artistic look. By using spoons and avoiding hand wash before and after lunch and snacks, we can save 40 ml per day per student. So we consider the 212 students with who bring spoon among 380. So we can save 84.8 liters per day and 17,808 liters per year. And it's a big amount. By adding aerators to the tap, before adding aerators to the tap, it took nearly 2 to 3 seconds to fill 250 ml glass, but after adding the aerators, it took 22 seconds to fill a glass. In short, we save water in the following ways. By using aquarium water once in 15 days and sending wastewater from the wash basin to the garden, by attending certificate staff, and by using spoons and snacks for, and I should be happy because they are resting and they are reading. Even their chirping have attracted many birds like the bulbul and sunbirds, but by the time we grab a stone to take a picture, they were gone. And we also have a small perimeter garden to attract birds. We have 2 guava plants and 1 fig tree to attract, so we can attract. We have only very small sets of things. One is Bahari of 320 square feet and one more is of 100 square feet. And this is the insect hotel which we need to attract insects from wooden boxes and some trees. And environmental educational programs. Our school supports many environmental educational programs. In that, eco club programs conducted by Mr. Gujarat by reduce the use of plastic. In our school, packet foods and water order due to constant awareness campaign, instead of fishing competitions or grant-making competitions, no thermocol is used. We don't even use thermocol for displaying diagrams or large diagrams. Current eco-friendly ingredients. All our programs are aimed at minimal waste generation. We don't use thermocol, we don't use plastic for project works. And in our best question papers has space for answers so that the project papers could be saved. More natural ingredients. To chase away mosquitoes, our tires wipe the grills of the windows with eucalyptus oil, which has a pungent smell. And we have kept camphor and paper cups in the corridor. We find this very effective. Our school has kept a paper incinerator. We burn the waste paper generated in this incinerator with less amount of smoke. Our the waste paper generation in each classroom and inform the respective class teacher if the amount of paper was more in the class. The others collect the paper and burn it in the incinerator.
paper and burn it in the and we can write the two subject notes in one notebook if the teacher is same. There's a picture of the paper insulator. Participation in environmental activities. Our participation in the symposium itself is a big sign that how much we value the environment. Our seniors have done many projects like water auditing, using waste air as a fertilizer, making biodiesel from cooking gas, etc. are some of these. There is a picture of the sanitary pad incinerator. Air quality monitoring. In the last symposium, our seniors have presented a project on air quality in classrooms. Since then, we occasionally monitor the CO2 level in classrooms. After the project, we have kept many indoor plants in the corridor to improve the air quality. National cooling system. Roots of these trees are in the cellar soil, and these branches were guided out throughout the mesh when they were young, and its branches reached to the first floor of the building. The same is done on the other side. It also gives a lot of shade. Conclusion. It's not about the space or the facilities. It is the commitment towards the environment to make us do things. As a student, we have learned many things in our school, and I am sure that most of us will carry this knowledge and will surely apply wherever possible. The positive impact would be unimaginable. Acknowledgement. We thank our school management, the teaching and the non-teaching staff, would be Dr. TVRAMACHANDRA sir, and his team of CES at IISC, Bangalore for
like birds, butterflies in your school campus. You have created green campus with lots of plants, indoor plants. Any activities? Let me spend those language. That's good. Any questions from audience?
Something like that. They always recommend. So do you still support that we should use spoons instead of our hands while eating?
See, if you go like this, anyone in our ancestors didn't recommend us to use plastics to spoil the things. So we kept it so clean and that spoiling everything. We just have to adapt and we just have to go.
So you are not recommending hygiene.
Yeah, you can see hygiene, you can have means like use sanitizers. There are sanitizers in our schools.
The sanitizer bottles also come in plastic bottles if you notice.
See, they can be recycled. They can be recycled. See, the plastic we cannot completely stop using because we have adjusted to it at that level.
Say, clapping with one answer, clapping with another answer. But what they recommended, that you have to think. What they presented, it matters. Because they are not telling you to don't wash the hand. But nowadays, all schools are okay. For lunch, okay. If you wash hands, it's okay. They are not told to wash hands, but reduce it. Okay, that's not the meaning what they are doing.
Hands quite often. We can wash, but not in a talking to our friends and leave it at that.
So you know that you avoid plastics in your projects and files by using, you don't use thermocols. So mostly projects you use papers, cardboards and all of that. And you said you have an insulator in your school, right? So insulator does not combust, it does not use oxygen to burn it. So that's going to cause a bigger problem, right? Because you're going to have a greater production, like use of papers. And then when you insulate it, you're going to produce more harmful gases. It's going to be carbon monoxide.
The gas produced from that is negligible, but the waste it is going to landfill is more possible.
Yeah, but then you're going to waste a lot of paper, right?
No, we are not.
The incinerator they're using to incinerate paper. Like, you have NGOs out there which some of them with pulp and they repurpose them with other things, right? We have recycled paper, now we have recycled straws as well coming up. So, why not? Is there a reason you chose the incinerator over these?
So it's just a suggestion. So instead you burn the paper that you collect in your school, you can give it to some small business like now they are running to make some paper pencils. So that you would reduce the carbon footprint that is generated from your school. making they will not use the. They take some charge for them. But it only takes papers. They recycle it. So to reduce the amount of carbon footprint, so this was a suggestion. Why would machines take?
To prepare a pencil, to hold it should be soft.
No, what they do is first thing they take the papers, they clean it, and then they.
So, right.
So my name is Anna sir, and I think everyone sure that it uses oxygen for its combustion and does not produce smoke. It produces white smoke which is eco-friendly and it does not harm environment. Thank you.
Okay, it was a code of, yeah, okay. And lunch is provided, morning where we had breakfast and night dinner. Netravati hostel. Netravati hostel. 20 minutes only. Please do have quick lunch. Fine. Now I request all of you to settle down. We'll start quickly. We have another 12 more projects to go. And certificates will be issued at last, finishing everybody's presentation. Please cooperate. Let's have a healthy discussion and not complication. Next team is by Niveda and Amrita Shri. Over to you both. We got a pointer, left, right,
One second.
Health issues. Our project is about creating a butterfly park with periwinkle plant. Introduction. Butterflies are the pollinating agents that keep the local species of plants alive. Butterflies of some caterpillar eat aphids, which are pests, and control their population. Also, caterpillars of some butterflies like monarch butterfly eats milkweed plants, which are weeds, and control their and therefore helps farmers indirectly. Also, butterflies and caterpillars play a most important role in the food chain. Increasing their population helps in increases of local predators, including birds, which leads to seed dispersal and plant pollination. This makes an ecosystem rich with its native species. Several beautiful creatures are becoming a rare sight, including butterflies, because growing a flower garden or vast plant of butterflies is not considered by many due to shortage of place and lack of awareness. We are trying to find out a way to increase the population of butterflies within limited resources. Objectives: to revive the habitats of butterflies, to understand the species of butterflies that get adapted to periwinkle plant, to create awareness about shrinking habitat of butterflies, to encourage students to
Why periwinkle? Periwinkle is one of the plants which is easy to grow with minimal care. These plants have high resistance against pests. These plants can be grown in most kind of soil. The germination period will be nearly 10 to 15 days. The time taken to flowering will be 60 to 80 days. Materials required: seeds of periwinkle, garden soil, seed, water. Butterflies are frequently used in that garden. The seeds we ordered on Amazon contained mixed varieties, so some of them we kept in the pots. The balsam plants could be seen among the saplings.
this Edbus macrops, which is also known as the common owl moth. The specialty of this moth is these are some moth and butterfly observed near our study area. Black-headed wasp, Melanitis leda, which is also known as common unique brown. We came to know that these butterflies were commonly found feeding on periwinkle, which is also known as Urema brigida. These are some photographies of other butterflies observed near our study area. Black swallowtail, lemon immigrant, lemon fancy. This is the python of butterflies observed by us. In total, there are 4 varieties of butterflies: mottled emigrant, lemon fancy, black swallowtail, lemon emigrant. In total of 4 varieties, mottled emigrant could be seen more. Conclusion: we will be able to conclude our observation only after the plants we grow in the garden start flowering and butterflies of Madagascar periwinkle, which is used in treatment of chemotherapy. Madagascar periwinkle and other Cataratus species in treatment for diabetes, hypertension and leukemia. Considering all the aspects, it is better to grow a periwinkle garden which develops the population of butterflies and also may check the growth of other invasive species, because
We have learned during the study that periwinkle shows invasive nature in warmer climates.
These are the references.
Thank you for your presentation. Can you please come back to those image slides? Yes, stop it here. And how did you get this common mind butterfly in Bangalore?
This was actually taken by internet.
Okay. You have put source also, but before quoting or taking information, you should cross verify whether that species is seen in Bangalore or not. Clear? Next. And which one is the below species?
Small grass yellow.
Cross check once again. This is not common grass yellow or small grass yellow. It's huge. It is lemon immigrant.
Sir, actually it was small, like it's been zoomed by taking.
Okay, but still we can make out because of those spots. Understood? That is why common immigrant is also called as lemon. You can see those veins. Okay, you should change the species name. Next. Okay, what did you? Okay, it was more common and abundant. Next. Okay. So periwinkle is exotic or it is a native to India?
It is exotic. Like in warmer climates, it's exotic.
Okay. So Indian scenario to our Karnataka, Bengaluru
Sir, India.
is native. Okay. Any doubts?
Birds are not only beautiful creatures but also indicators of environmental changes.
Yes. So this is the picture of the birds we identified. There are the details of the birds we identified. And the register the details of the birds we identified. And the register bulbul, which was a frugivore. In IISC we found these birds in which almost were omnivores except crow, which was a scavenger, and kite, which was a carnivore. In Madhya we found these birds in which two were carnivore and one was omnivore. In Bangalore University we found two birds in which one was opportunistic omnivore and one was omnivore.
In Fire we found this bird in which almost everything was omnivore except red-whiskered bulbul, which was frugivorous, and white rooster, which was insectivorous. In Nagarjuna we found peacock, which was omnivore, and red-whiskered bulbul, which was frugivore. In Jayamaha we found intermediate egret, which was carnivore, and Indian spotted predator, which was omnivore. Out of 16 birds, one was frugivore, 11 were omnivore, 3 carnivore, and 1 scavenger. Sighting more omnivorous birds indicates that they are of higher adaptability.
So these are the birds we identified. This is Indian spotted billed duck. This is the intermediate egret, white rooster, Indian robin. This is the Asian coil, red-whiskered bulbul, peacock, jungle myna, house crow, feral pigeon, blue rock pigeon, the small Indian kite, greater cowcurl. This is the tree pipit, rose-ringed parakeet, and this is the house crow, house parrot.
Summary includes the IISC activity. It can be assumed that the urbanization has chased many birds out into the wild, and the place has been taken by more adapted species. For example, red-whiskered bulbul is in great danger due to the loss of habitat, whereas red-whiskered bulbul is more adaptive. Sighting more omnivorous birds than frugivorous bird is planned to find out what kind of plantation can improve the useful local birds population. All the details about the native plants and native birds must be identified here. As the students, we can acknowledgement. We thank our school management, teachers, and friends for helping us with the project. We also thank our parents for encouraging us. A special thank to Vijula sir for being an inspiration to study about the birds. We sincerely thank Dr. TVRAMACHANDRA and his team of CISC department, IISC for giving us an opportunity to present the project. Some of our friends, Elizabeth, Manishri, and Gita Gowda, have greatly given
Thank you, Kubra and Manisha, for your wonderful presentation. Can you please come back to bird species, bird images? This is Indian spot builder, correct? Next. This is also right. Which bird do you think? It is pied bushchat. Pied means black and white. So please cross verify once again. Next. This is Indian normal, correct? This one. Is it a coil with such large beak? No. Please cross verify once again. Red-breasted bulbul is correct. Peacock. This one, Bengal one is correct. See, this has there are like two crows what we see. If it is completely black, Bengal crow. If a neck is grey, house crow. Grey, it had grey neck, is it? Okay. Next, feral pigeons, blue rock pigeon are same. Okay, it is only subspecies. So subspecies will not consider as a new species or addition. It is like both are same. So one is -1. Black kite, okay. Later governor, correct. Yes. Please come back. Who can identify this bird? We already had a presentation about this bird species. Yes. Baya weaver bird. Okay. These are Baya weaver bird and not the repeat. I request all of you to send these kind of like once you prepare a draft, send it to the experts and get their consultation regarding species confirmation. And this is Rosemary parakeet, house sparrow, correct? And how did you get this IUCN status? When you are seeing, you should also cross verify because Bengal men are least concerned. They are not nearly threatened. They are least concerned. Next. Even our parakeets are also least concerned. Their population is really good in numbers. Okay. These two you will have to change and cross verify. They are not least concerned. They are not nearly threatened. They are least concerned again. Next. Any doubts from audience? Yes.
What do you mean by frugivores?
Birds which eat fruits, which they depend on fruits more.
Why aren't they called herbivores?
Because they don't depend on fruits only. They just eat fruits. They depend more on fruits.
Herbivore is like full of green plants, leaves, grass, and all edible parts of plants. Whereas frugivores, their special diet is only related to fruits, berries, cherries.
You told that if a bird is turning into an omnivore, it shows better adaptability. But then later you told that omnivores are greater affected by climate change than herbivores.
Yeah, nowadays we are cutting more plants, so like frugivores birds, like they are not more. Omnivores they can depend on anything. Frugivores like they need more plants, fruits and all. So like we are citing more omnivorous birds, then it proves that we are saying more omnivorous birds.
Okay, so frugivores are less more concerned.
Yes. It happens when birds fly from, like they migrate. So yeah.
His question was that good food yielding trees. They also feed on insects. If good insectivore birds are seen, that means there are a good number of insects population. So each food diet, their behavior and also the habitat preference indicate certain birds which are endemic to scrubland or forest land or wetland birds. Any other questions? Okay.
Tanishri. This is my partner Elizabeth. We are studying in grade 10 in Bangalore Girls High School. So our project is about Parthenum Insteroferus. Parthenum is a poisonous and aggressive weed. We have noticed weeds like Parthenum and Echinacea growing widely around our surroundings. The role of invasive species in destroying our native species. To identify why Parthenum Insteroferus is growing so widely and rapidly, to experiment various control measures, and to find possible ways to beat and control the population of Parthenum. And in our study, we are required our neem leaves, case study plant which is Parthenum, white neem leaves, water bottles, logbook and pen, neem seed, garden soil, seeds and gloves. Preparing neem extract. We prepare neem extract at our homes, and for it we took nearly 300 to 400 grams of neem leaves and boiled it in 1 liter of water. After the color changed to brownish green, we filtered it and stored it in plastic water bottles. Applying neem extract on Parthenum plants. Our study area was outside our school campus, adjacent to the exterior wall. On day 1 and day 2, we observed no visible change. We noticed Parthenum beetles, also known as Mexican beetles, on the plants, but no considerable damage were noticed by these. On day 3, some of the leaves were wrinkled and shrunken, but we were not sure whether it was because of the neem leaf extract. And on day 7, more leaves looked very twisted, and plants looked unhealthy, and some flowers appeared to be withered. Day 12, the leaves started to turn yellow and they kind of looked unhealthy. Even the flowers started to turn brownish. It was on day 15 that we noticed major changes in the plant. Visible in the adjacent side looked the flowers looked very bright white in color and the leaves were green. So on day 18, most of the flowers withered off and the plants looked very weak and yellow in color. We wanted to know if neem extract is causing any problems to other plants which is in the surrounding area, but we found the other plants healthy. Day 20, the plants show that they are dying. To again make sure of our observation, we apply neem extract on a baby Parthenum. The baby Parthenum showed signs of withering within a few days as compared to the fully grown plant. Hence, our study turned out to be positive, and it shows that neem extract has a significant effect on Parthenum. Day 22, we increase the concentration of neem extract. Initially, we boil the leaves as it is. Later on, we mince the leaves and then boil them. Overall, the plant. On day 1, this is how our plant looked, and after 30 days, you can see that the plant looked very weak and it showed the signs of dying. We were worried, as I mentioned earlier, we were worried that neem extract might cause any damage to the other plants which is next to the Parthenum, but we found the other plants healthy. And since our experiment proved that neem extract played a significant role in damaging the Parthenum plant, we wanted to take the study further, and hence we did neem seed balls. A special thanks to the students of 8th standard who shared the filter garden for our project. We tried to grow. Conclusion. So Parthenum being a strong weed has no effective natural predators. However, concentrated neem extract can check the growth of these plants. We also found out that azadirachtin in neem has weedicide property to control these invasive species. Initially, we applied diluted neem extract, so the results were obtained slowly. Propagation of neem plants can also check the growth of these invasive species, but further studies are required. Propagation through seed pods is a slow process, so instead we can directly sow the seeds. We found these plants growing vigorously in empty site, so the site owners can be held responsible. Recommendation: instead of using chemical herbicides, we can use neem extract, which is an eco-friendly and safe option to control these invasive species like Parthenum. We further wanted to continue our studies, but during the last days of observation, the VPMP removed our Parthenum plants. However, manual removal does not ensure that the plants won't grow in the same region again. Since their large seed producing capacity is helping in the fast colonization, we need to ensure that the seed producing capacity is effective. And this is where we want to draw your attention. These plants are growing widely because of the flower. Possible is also to students in schools, so that they can check the surroundings and find control measures to control the population of these species before they spread. Acknowledgement: we thank our school management, teachers, and non-teaching staff for cooperating in our project. Our sincere thanks to Dr. TVRAMACHANDRA Sir, CES Department, IISC,
Bangalore and his team for providing us an opportunity to present our project. We also thank our parents for their encouragement and support. These are some references. Thank you.
Thank you for a wonderful session. It was really good, neat, small, approachable, doable, economically viable, and environmental friendly. Apart from Parthenium, have you tried your neem extraction oil on any other plants?
No sir, because in our surrounding we found only neem.
No bonus for you. Same person asking multiple questions.
Hi. So in your last few slides you mentioned that manual removal does not guarantee that it will not grow again. So
Using neem or just putting it out like that. Can't you safely put it out and with the soil plant it somewhere else? But we think it is having major harmful, it is doing major damage to the environment than the medicinal properties what it has.
These Parthenium get pollinated?
So this Parthenium is different. This is wind pollination.
Any other suggestions or queries, questions? After this, Mahi and Nityashree please study. And then Shreya, Hemanya and Parinita. You are also here.
We happened to hear about a farmer, Sri Kumar of Wayanad district, Kerala, who grows some about such dedicated farmers who want to preserve these varieties of paddy. He is the recipient of many awards for his effort to try these medicinal varieties of paddy to keep alive across India. It is very essential for us to know the materials required there, varieties of 10 seeds and saplings of anduri, pots, garden soil, wooden spoons, marker, logbook, pen and bottles to water. These are the images of planting anduri saplings and these are the 10 varieties of seeds. These are the images of sowing seeds of 10 varieties. Observation after 5 days. Since the anduri saplings appeared right, we contacted the farmer. On contacting to the farmer, he assured us new saplings took sprouts here and there. Since the plants did not grow as expected, we called the gardener to sow fresh seeds. And these are the images of sowing fresh seeds by gardener. One of our objective was to check whether the obtained varieties can be grown in controlled condition. We think we got a positive result. Features of the selected varieties are, first one is anduri. It grows up to 5 to 6 feet in height and since the name suggests as the color, it is of black color and its duration is 150 days. Thus medicinal value of this is rich in antioxidants and good for diabetic patients. It is also rich in fibers, vitamins and minerals. Next one is Gandhalashala. The place of Gandhalashala is rural countryside of Wayanad, Kerala. The specialty of it is it is aromatic rice and was grown by tribal people. The medicinal value of this is it is diabetic friendly. It has very low glycemic index. Kunjan Thundi. The native place of Kunjan Thundi is Wayanad, Kerala. The plant is short saturated, hence it is named as drop. The medicinal value of this is rich in fibers, vitamins and it has very low glycemic index. Krishna Kaumur. The native place of this is Gujarat. It was grown by the tribal farmers of Gujarat on the mountains with backwaters. The specialty of this is it is highly aromatic and medium size. It is also known as Basmati of Gujarat. It is great rich to maintain electrolysis. It also has nutritional values and essential for diabetic patients. It is also diabetic friendly. Next one is Navara. The native place of Navara is Palakkad, Kerala. The specialty of that is it exists in two forms, gold and black color. The black color Navara seems to be healthier when compared to golden one. It is used in Ayurveda as healthier food It is also used to treat diseases like arthritis, paralysis and it is good for rejuvenation also.
Ramli. The native place of Ramli is Punjab. Speciality of this is the seeds are blackish brown in color with white brown wool in outermost layer. Medicinal value of this is rich in source of vitamin E and antioxidants. Next one is Jeera Kashala. The native place of Jeera Kashala is Wayanad. The speciality of this is famous for its characteristic fragrance and aroma. It is highly disease resistant. Medicinal value of this is easily digestible and good for the diabetic patients. Next one is Dabar Sela. Native place of this is Maharashtra. Speciality of this is long grain creamy white colored rice. Medicinal values of this is abundant in fibers, vitamins and minerals and has low glycemic index. Raktashali, the native place of Raktashali, Dakshina Kannada in Karnataka and Kasaragod in Kerala. The speciality of this is the rice has distinctive red hairs and grain. The medicinal value of this is rich in antioxidants, ions and it is used in Ayurveda to treat three doshas, Vata, Pitta and Kapha. Next one is Chengalu. The native place of this is Wayanad. Special features of it is heir to rice handed down by the several generations and it is planted by the several like generation by generation. The medicinal value of this is used for treating in diarrhea and vomiting. It is used for recovering from jaundice. Why these varieties were ignored means most of them are of long duration and tall trees. For example, if we take Chengalu, which is one of our selected seeds, it takes about 120 to 125 days, whereas the duration of Falakaramata, which is commonly grown in Kerala, takes 90 days to grow. Similarly, the duration of Jeera Kashala rice is 150 to 180 days. Moreover, yield per hectare also varies. For example, Gandhara Shala rice is long duration variety with all tall plants and have very low grain yield. For example, it gives 2.0 to 2.7 yield per hectare, whereas Sona Masuri, which is commonly cultivated in Karnataka, gives 6 ton per hectare. Conclusion: there are very small set of farmers who try to keep these indigenous varieties alive. We should support them in all possible ways, and people can even consider them in growing in the controlled condition, and they can have a healthy marketing, and we can reduce the allopathic medication wherever possible. One of our objective was to check whether the optic varieties can be grown in controlled condition. We think like we got positive results. Recommendations: Agriculture Department can take a serious note on preserving such indigenous varieties and recognize the farmers individually. The effects need to be recognized as they are doing a wonderful job by helping mankind by preserving the native varieties and maintaining the gene pool. We strongly believe that our science textbooks should contain some topics about this so that students will know one positive option in their future. Acknowledgement.
We sincerely thanks Dr. TVRAMACHANDRA sir and his entire team of CES and IISC for giving us an opportunity to present our project. We thank Sri Prasid Kumar who gave all the necessary informations and seeds and targets. We also thank our principal for giving us all the assistance to conduct our experiments in schools. We then thank our school security who always accompanied our school us to the terrace for monitoring and taking photographs.
These are our references. Thank you.
Do you know any rice variety which can be grown in a sea water or a brackish water?
Our motto was to check whether they can be grown in controlled condition. So we found a positive result, but we did not check with salty water.
But is there any rice variety which can be grown in the sea water or brackish water?
We are not aware of that. It might search.
In Udupi, I think they can. Udupi is there.
We'll check.
Yes, thank you for your wonderful presentation. It was really nice to hear about rice. Any questions from audience? Only one question. College.
As you guys said, most of your plants
because they are slow in giving it and they have less yield. So not completely should be replaced. 50% can be replaced so that we can shift from less nutritious food to highly nutritious food.
5 minutes. Trying to have summary from Nishan. He will summarize for us what you both have presented. Can you please summarize? Thank you so much. All the best. Next, Mani Aunty.
by putting used soil pads in flushes, and this leads to clogging the drainage pipes. So it is not mentioned here, but I would like to inform that in India only 1.4% people use modern menstrual methods. Menstrual cup 1.3%, tampon 0.1%, majority 98.6% use sanitary napkins. In that, 96 people are non-biodegradable. They break down into microplastics and thereby contaminate soil, water and air, etc. Health hazards of open disposal of sanitary napkins. When soil napkins are burnt at low temperature, they release carcinogenic gases and dioxin fuels. The disposed soil pads are an eyesore and it destroys the beauty of our nature when it is flushed down and therefore clog the drainage pipes. The household hazard waste is proposed to be collected separately by BBMP. In reality, BBMP workers collect all the dry waste together and they send these to the nearest common biomedical treatment facility and incinerators. Estimation of sanitary pad waste generation in schools. So, the total waste, this is an estimation of total waste generated by school girl students alone in Karnataka. So, approximately 11,27,385 students are there, and the waste generated by them is 81,17,172 kg, that is 8,117 tons. So, what if we could make it zero? These are the survey we did manually. These are the survey questions we have done. Survey and awareness camping at Government High School, Cottonpete. We did a awareness camping at Government High School in Cottonpete. There we got to know that there is no much awareness regarding the problems created by improper, we were 150 to approximately, and the pads disposed along with school waste was zero. So, utilization of sanitary pad incinerator in our school. So this is the incinerator in our school which we have. We took demo and the ash which was produced by the incinerator was negligible. Comprehensive report of the survey done manually. We even conducted a survey through manually. Total 35 members participated in that. The waste generated was used infections. 70% mentioned that they dispose the pads wrapped in paper. Here 70% mentioned that they would like eco-friendly disposal methods if provided. 53% mentioned disposal methods from schools and colleges. Only 6.7 cloth napkins, and with the help of Vijayanagara Club, I would like to inform the advantage of cloth napkins that it can be used up to 2 to 3 years and it can hold up to 4 to 5 hours. So it is the best. Reusable cloth bags create issues of changing the pads by the
public place. Incinerators can be the best alternative for disposal. The gas generated is negligible and can be flushed down once in a month. Smoke generated is a concern, but considering the volume of solid waste generated and other consequences, incinerators are the best option. So this is the Palakkad Municipal Awareness Camp acknowledgement. Our sincere thanks to Dr. TVRAMACHANDRA sir and his team for providing us opportunity in our survey. Thank you. These are references. Thank you.
Okay, thank you for your presentation. Any doubts from audience?
The audience woke up. Congratulations, girls. I was really impressed the topic that you chose and how comfortably you put it across.
Does that happen? It means, see, the used napkins, when they are dumped in landfills, they spread various diseases. So that is evidence.
Okay, so can it actually get to such a level that it spreads all across the country? I mean, to an epidemic level?
Yes, it will lead you to dump the used napkins in landfills.
Lovely presentation. I just had a doubt. So the incinerator that you mentioned, the incinerator you mentioned, I looked it up and found a couple online, and they use usually 230 volts and run on 15 amperes. So this is approximately 3.45 kilowatts of electricity. So meaning the energy consumption is justified now. This is the same as 2 to 3 residential air conditioning, right?
It matters of hygiene. As she said, it causes many disorders. So as you know, human health and their life is way more important than the resources you use. So I guess that is a odd.
So you told that you have to go backwards in using the cloth pads. First of all, using cloth pads to a duration like 4 hours with normal plastic pads are very harmful because the bacteria which grows. And ಅದಕ್ಕಾಗಿನೇ ಅಲ್ವಾ ನಾವೀಗ ಪ್ಲಾಸ್ಟಿಕ್ ಪ್ಯಾಡ್ಸ್ ವುಮೆನ್ ಇನ್ವೆಂಟ್ ಮಾಡಿರೋದು. ಈಗ ನಾವು ವಾಪಸ್ ಅದು ಲೈಕ್ ಕ್ಲಾತ್ ಪ್ಯಾಡ್ಸ್ಗೆ ಹೋದ್ರೆ ಅದು ಇನ್ನೊಂದ್ಸಲ ಆಗುತ್ತಲ್ವಾ. ಸೊ
See, in when our ancestors in those generation, they used to feel so shy. Yeah. So they used to feel so shy, and they were not expressing what they are feeling. And those what they used to use that clothes, and they used to not. They were they used to not to dry it properly, and they were not even washing it properly. So that's the reason why it was unhygienic. But nowadays it's not like that. So there are more cloth pads available, which is.
Can you please tell me what is the percentage of people who use specific things of menstruation items, because I want to know.
Yeah, menstrual cup is used by 1. So in India only 1.4% people use modern menstrual methods. In that, menstrual cup is used by 1.3%. And tampons 0.1%, and majority 98.6% use sanitary napkins. In that, 96.3% use disposable pads, and 1.4% use reusable pads.
Where did you get this data from?
In internet.
Okay, thank you for your wonderful session. We'll move to next session. Thanks to you. All the best. As ma'am said, we should really appreciate for what they have taken topics. Huge round of applause for their bold and brave to take or pick up such sessions to create awareness on this huge platform. Next session, Shreya Aunty.
A very good afternoon to all. It's a true honor to be here among all the esteemed experts, passionate environmentalists, and everybody who keep a deep commitment towards preserving our nature. Our environment is not just the backdrop of our country, but it is a true foundation of environment, community, everything. Bangalore, a metropolis city. Besides it, beneath its diverse and dynamic soil, there is a dynamic soil.
You can see.
No, sir, they'll do it.
And after that, Shresha, that team is here. Aishwarya, that team is also here. Team is here. We have last 4 more presentation. Normally we would like to wind by 4. Okay? Cooperate.
What's your name?
Third one. You are ready? Once we start. Slide center is here. Left and right. Center on this point.
A good day to everyone and the adaptations to our local conditions, exploring how our native fruits have medicinal and ritual connections to our traditions, explore how market native and exotic fruits and assess environmental impact. Jamun and Amla have similar nutritional values to blueberry, kokum and bilbim and so on. So here are the first one has a few nutrients, exotic fruits, native fruits and a comparison. So we can see that jamun and so here so according to this table we are comparing the distance covered in importing these exotic. This is the graph. So these graphs and of this slide and the following slide show how the import of egg from there to the main hub of Karnataka like KR market, from there the distance calculated and the carbon emitted from the traveling of avocados 600, and this is a comparison of like how the native fruits which are grown locally or in different parts of country. There are a lot of chemicals and preservatives used on them, so which is edible wax, calcium chloride and sulfur which these exotic fruits go through so that they don't spoil over the journey. And these processes and preservatives cause these and how much native fruits they sell. Like in from a local fruit vendor, one is season at one is returns. As we can see, most of them sell the seasonal or the occasional native fruits which are most commonly used for the festive seasons or for the daily consumption of.
importance of native fruits in India. Bael is known for its digestive benefits and is used in Ayurvedic treatments for gastrointestinal problems. Most of its parts, which is leaves, roots and bark, are used in medicinal applications. And also, Bael holds a cultural significance in Hindu rituals where it is used in Mahashivratri, and its leaves are offered to Lord Shiva, symbolizing purity and health. Jamun is valued for its ability to regulate blood sugar levels. And amla is a rich source of vitamin C and antioxidants and so on. And so let's go to the observations. According to our observations from the research and our surveys, the amount of carbon emitted is more during the transport of exotic fruits compared to the native fruits. Buying regional fruits would be an economic support for local farmers who don't depend on growing exotic fruit, reduces global warming.
And our recommendations, which everyone will
this opportunity and letting us showcase our talent over here. We would like to thank our teachers and staff for helping in the research work. Thank you.
Like the wild fruits, which
River and Nihal team. Srisha, Vibhar and Nihal. After that, Pratik and team. Will you accept new PPT? They have made some slight changes in it.
I already confirmed that. You mentioned
What are the values?
the values are very
You are ready? I will set your time. What time? Off that. Switch off that.
that is directly. You get directly connected.
So hello, my name is Nihal. This is Shrisha and this is Vibhor in public school. It is an area of 27,472 meters square and 807 a perimeter of 807 meters.
So our main objective here is to identify all the tree species of VPS campus and to measure the carbon sequestered by all the trees. We calculated how much amount of carbon is sequestered in how much area of all the trees. So the methods, there are actually two methods which we can do, direct and indirect method. Direct method is very harmful, we should kill the plant, but that's more accurate, we didn't do that. It gives us AGB. By below ground biomass is 0.2 into above ground biomass. TDW is total dry weight, 0.725 into total biomass. So at this point we should know 72.5% of total biomass is dry weight of the tree. So to get the total dry weight is 0.725 into total biomass. The remaining is moisture. Total carbon weight is half of total dry weight.
Amount of carbon sequestrated by the trees, area occupied by the by all the trees in our campus. Then we compared the rate of carbon sequestration with the area, which gives us the rate of carbon sequestration of a school in 1 meter square. This is the all data which is being collected.
You explain.
Is it a table? I just need to click on next slide. It is not visible over here. It is a blank. Okay, explain what is it? This table is there. So you can explain this.
So in the result section, which is not there. So at the result, we found out that the highest species of trees which are more carbon sequestrated was teak. Teak is 29%, 29.3%. Then comes at the second place, which is coconut, which comes at 19%, 19.7%. Then comes the Ashoka tree, which 10.3%.
And 466 tons of carbon sequestered by all the trees, which is about 16.9 kg per meter square. So that is the conclusion that we reached.
These are the references.
Above ground as well as below ground, right? So why you guys didn't collect any values for below ground? Below ground is there. Can you show me? Like, and you took the total ecosystem value, or like you added and that?
So the last conclusion you just do, conclusion the company, the 316 trees which have a carbon sequestration of this terms. Can you explain in terms of a ton? How much ton? So in 1 ton is equal to how much kg?
You can also mention ton per hectare value. In a hectare, how much?
We had initially taken the area in meters square.
That will be better.
They are going clockwise, it is fine. They are
No,
going to the trees.
they can take the random thing out. I think they had the value, but it's okay. Just suggestion.
When you mention like in conclusion with respect to tons, no, so that gives the various mass balance of it, so that will give more. Any other is there? So we'll move to next. Aishwarya and T.
Sir, they showed they are ready now. So we can do it. They are ready. So they want to discuss on that.
After last. We'll last will do.
Last.
So Aishwarya and T, please come.
Followed by Pratik, and later you can present for commenting.
The objective of our study is to compare the soil quality between the soil present near the banks of the lake and the school garden. We also intend to analyze the quality of the lake water and compare it with that of the borewell water which we've taken from the school.
Soil and water are vital for environmental health. So this helps us, analyzing these quantities will help us assess nutrient levels, detect pollutants and to monitor ecosystem health.
So this is our first study area, Pamkata Lake. The latitude and everything is given there. This is our second study area, Vidyaniketan Public School, which is our school. So our study is divided into two parts. The first part is soil analysis. Soil analysis is basically testing the soil for various parameters to determine its quality. So this by calculating the percentages of sand, silt and clay and plotting them on the soil pyramid to determine the texture of the soil. So next we have done the pH test, which basically tests the alkalinity or acidity of the soil.
So the three major minerals present in the soil are nitrogen, phosphorus and potassium. We have measured these minerals through the soil testing kit provided by the. Next we have done the electric conductivity test. Electric conductivity measures the concentration of soluble ions, primarily salts in the soil. So as per the values we have got, we can tell that both the soils are non-saline in nature.
So next we have done the TDS test. So TDS basically measures the total concentration of the dissolved substances present in the solution. It includes
given salts as well as dissolved organic matter. So according to the values that we've got, we found that the soil is slightly saline in nature. So the next part of our study is water analysis, which is basically testing the water for various parameters to determine its quality.
In that first we have measured the temperature. The temperature affects the aquatic health, the oxygen levels as well as the pH test of the water.
So next we have the calcium hardness, total hardness, chloride and fluoride test using the water testing kit which was provided by our school. And next we've done the electric conductivity test for water, and the EC value was recorded in microsiemens per centimeter.
Next we have done the TDS test, and which was measured in milligram per liter.
So next we've done the dissolved oxygen test. Dissolved oxygen is basically the amount of unpaired non-compound oxygen which is present in the water. This is very important as it helps us to determine how healthy the aquatic life is. We've done this through titration. So now let's go through the results and discussions of our studies. So first, coming to soil texture, based on the percentages that we've obtained, we can conclude that the lake soil sample has a sandy loamy texture which provides excellent drainage, good radiation and moderate nutrient retention capacity. And the school soil has a loamy texture which is a balanced texture. It has good nutrient retention capacity and good aeration.
Next, as we have done the pH test, the values we have got, we can conclude that the soil is alkaline and there are chances of nutrient imbalance and reduced microbial activity. After that, we have done the NPK test. As per the values, we can tell that the phosphorus and nitrogen content was slightly higher in lake soil compared to the school soil.
So next we've done the organic matter test. We found that the percentage of organic carbon in Kongata Lake was 1.25%, which is below the permissible limit, and in the school garden it was 4.13%, which is within the permissible limit. We have calculated this percentage using the formula here. Next we've done the DO test. We found the DO of Kongata Lake to be slightly below the permissible limit. We've also compared the dissolved oxygen of Kongata Lake in 2009 and 2024. We can see that there is a slight increase in the DO.
So here are the results and discussion of the water analysis we had conducted. First, as the pH, both the Kongata Lake water and the school garden water was alkaline in nature.
And coming to the total alkalinity, we can see that both the values are well within the permissible limit, but we can see that the school garden has a little bit more total alkalinity than the Kongata Lake.
After that, we have done the total hardness, calcium hardness, chloride and fluoride test, and all of them were within the permissible limits.
Then we've done the EC test. All the values are within the permissible limit, and we've also done the TDS test. For this also, all the values are in the permissible limit. And the dissolved oxygen, as discussed before, it is slightly lower than the permissible limit. So here we have compared the Kongata Lake water quality of 2009 and 2024. Here are all the graphs.
So as we have done soil analysis helps us monitor pollution and ensure compliance with standards, while water analysis helps us to prevent waterborne diseases and maintain the water quality.
So each experiment comes with its own challenges. The challenges that we faced in soil and water analysis included in sampling errors, equipment calibration, human error and complexity of results.
So as per the NPK test, the phosphorus and nitrogen content was slightly higher compared to the school soil, and then the organic matter was also comparatively low in lake soil compared to the school soil.
It is essential to balance out the nitrogen and phosphorus content to avoid situations like excessive algal blooms. This can be done through addition of organic matter to the soil, which will also correct the deficiency of organic matter which is found in the lake soil. We can also balance out the phosphorus and nitrogen through appropriate water management techniques.
We have tested the soil and water through various parameters like the pH, hardness and nutrients.
Our results are support the good decision making and effective management practices. This is the bibliography, the references that we've used. We would like to thank the Lake Symposium team and TVRAMACHANDRA sir for this opportunity and platform to work on. We would also like to thank the VPS schools, the teachers as well as the lab assistants who helped us throughout the process. And thank you.
Come back to the slides. Stop. While explaining, you have taken the word as 2009, but here 2015 versus 2024. Okay, no problem. Where did you put all this data?
This was found on the IISC website.
Yeah. Then all the experiments you were conducted in your laboratory. So based on comparison results, you are going to say that the X-based sand was good compared to lake.
Sir, can you repeat this?
You have tested two soils, right?
Yes.
Which one? One is
One we have tested
garden
and the lake soil.
lake soil. So comparatively, garden and lake soil, your garden soil is good. As compared to?
Yes. Garden soil supports better anthropogenic plants. We need the ones that we use in our daily life in class.
The word is anthropogenic presence both.
Thank you. Any questions from audience? Only one. Your mom has.
Why your school water have less BO content anymore?
Because the school water is a borewell water, so since it is underground for so long, the oxygen is absorbed by the soil.
How you guys collected the water sample for DO?
For the school water, we took it from lake. Lake sample was taken from the lake, and then we took the school sample from the tap water, and then we
by using which there is specific BOD bottles you should use for collecting DO, otherwise the values will vary. So
the samples were collected in glass bottles.
Okay, so the air bubbles will be there, so it can affect your values. So it will vary. That's it. Just a suggestion. Next time, please don't use like that. You get specific BOD bottles. You can use that for the experiments.
Thank you. Thank you.
Next, Pratik.
Last but one. Last but one.
You need any help?
School studying in class 11. So today I am here to present on the topic monitoring the lake using AI, and my objective is to monitor the lake's health with the help of AI, and also create a model which will help me to monitor the lake. So moving on, let's get back in time. So during the period of 1970s, the lakes were generally clean and they had a very great biodiversity. But between the periods of 1980s and 1990s, due to rapid urbanization, pollution, the lakes lost their habitat. But if you come to 2000s, the lake were completely worsened due to the more pollution, and as a result, the lake size were being smaller. So the percentage of the dissolved oxygen percent came down to 35%. If you look at this, the factors causing the water pollution, you mainly see that industrial discharges, agriculture runoff, municipal waste, and etc. Because of this, because the industries or the municipal waste, they are not treating the waste properly, it is major problem for the lakes. So this is Belandur Lake, and as you can see here, this lake was this huge from here till here. But here, here they have constructed some buildings, and also here it's decreased the size of. Most important thing in this lake to bring up the lake's health, to restore the lake health, with using the real-time monitoring data, and this data can be only found when there are sensors in the lake. So one of the sensors which I brought is pH sensor 452C. The 452C is the model name, and which is connected to the ESP32. ESP32 is the microcontroller which helps in most of the IoT projects. IoT is nothing but Internet of Things. So quickly let me connect this and show to you. Before that, I'll just introduce to my model. It is called ALTS, ALTS. That is nothing but all type of sensors. This all type of sensor can hold up to 5 sensors or more, depending upon the weight of the sensors, the connectivity and all. These sensors will also have an flora and fauna sensors around it, so that while it is moving in the lake water body, it does not harm any animals or the plants. Because we are talking about saving them, and if I deploy the sensors in the lake and it harms them, there is no use of it. So let me quickly show you. Here I have 3 solutions. One is acidic, that is lime water. A little bit I have diluted it, and one is soap solution, and another one is normal water. So I'll just connect and show it to you.
You can ask any one of them as a just to observe.
Okay, I would like to invite my friend Nihal to come and look at here.
Not your friend, you ask anyone else, so they can believe like it is.
Okay, who would like to volunteer? Just raise your hands. Volunteer. Anybody else? 6.9 to 7.1. That is the value. So when I dip it into other solutions, it will show. So I have ESP32 which will take approximately 5 volts. 5 volts which can take, so that is the reason I have connected to laptop. You can ask one more question, why laptop? When I deploy into water, I don't have laptop.
Next, Shreya and team is ready. Yeah, PPT is over.
So that's 6 minute time. Time is over. You can finish your PPT presentation is finished. Then
Okay, so here are the cost of the sensors. Okay, see if you see at water flow sensor 240 rupees and all etc etc. This is the 5 basic sensors we need for this, and I have taken this. Okay, it's also included with the installation charges, maintenance charges, everything. And also if I purchase any other link gets damaged, we can use this real-time monitoring data, so that we can fetch the data and use around 2 lakh, 3 lakhs rupees. I think that should be enough for the government. If they don't invest this much also, I don't know what to tell. So moving on, these are the prototype images. So my model, I thought I would design. I don't know why it's not working. Before that, I'll just show you my website. So this is my website. This website name is Lake Monitoring, and also bro, you just see what you see.
Lake right now. Also lake.
This project will be implemented by the government, or when I earn enough money to implement this. And just contacting details and all there. I have taken 7 lakes. 7 is a good number, I feel, Ronaldo. So I have taken it. So there are 7 lakes, and I have done that. When I click on that, there is a lake image about that, and few informations of the lake. And in the home page, you have the what you call. See, in the home page, you have the lake health index basically. Next presentation, I'll just connect it to internet and I'll show the pH values that it's working. Okay, just
you can put the link of your. You can share.
So that's what I'm telling. I have not posted the website. It's just a zip file.
Okay.
No, we'll continue the next presentation. It will take some time.
Yeah, I'll keep it there and
meanwhile, other presentation now.
We have last presentation.
Yeah. Please come.
I think please come.
Okay, so good afternoon everyone, and sorry for the messed up part that time. I just wanted to reach myself with my friend and my brother. So yeah, so starting with our presentation, I am Shreya, my friends Hemanya and Kalinita. So our topic is urban agronomy with soil analysis of West Bangalore. So as you all know, to agriculture and environmental protection, understanding these hidden treasures is key, and that's what we are doing now. So, moving on to the objectives of our project, it is to identify and analyze the physical and chemical parameters of the soil and to find out various crops that can be grown in these type of soil. And finally and most importantly, it is to promote local soil awareness, to grow valuable plants, reduce transport carbon footprint and minimize spoilage, and support sustainable urban living, Rajarajeshwari Nagar, Ullad and Kengeri. These are the locations and the longitude and the locations we have given it here. So, coming to the tests we prepare, we have bulk density, electrical conduction and table. Ullad soil, we have taken 2 samples of each locality soil. Bulk density, we have got it as alkaline, and NPK. NPK's normal range is
4:2:1. But here all the soil we have got potassium. But here all the soil we have got potassium. The range of potassium is high in each soil. Soil that we have tested are all around the western Bangalore is alkaline in nature. Moderate to high levels of dissolved is more than the normal range. So in this type of soil, what are the plants that can be grown? So in this type of soil, what are the plants that can be grown? So the plants that can be grown gardens, particularly vacant lots, biodiversity. And we have often seen our elders at home doing rooftop gardening as they think it's a way to connect with nature, and they also contribute to the soil. And that is what inspired me to do this. So I would like to thank the IISC Institute for giving us an opportunity. And I would like to thank my school and my guidance teachers and also my friends who supported me throughout the journey, and also today. I am very thankful to them, and thank you all.
Selected where Bangalore is your study site?
Yes, sir. From our childhood like West Bangalore, and we have seen the city grow from day to day. We know from where surroundings was, and then explore more.
Rajeshwari Nagar is like it is from the very old time, it is the area is there. So now like urbanization has impacted its work.
Okay, round of applause for that. Now Pratik is with us.
Sir, I would like to call upon that boy who was here. So then, Shaurya, can you please come? So, yeah, as I told you, this pH comes with a tube. So if you could also come here. Yeah. So a pH comes with a tube which has a very.
Can I connect my data? You got data right now.
So yeah, you can connect, but I have to upload the code again. It will take a lot of time. So yeah. So yeah, as everybody can see here, this is water. What I have told. Okay. Sorry guys, I put it basic solution. Sorry, I was shocked. Sorry, I'm very sorry. So you can see it is. Yeah. So it's 8.5. 8.5. I think we can consider it the base. Yes or no? Yes. Okay. Very good. I use the soap water. Okay.
So it shows the value 4.97.
Okay. Can we consider that as acid? Yes. Okay. Very good. Okay. Let's test it for the Alva's purifier. Okay. We'll check the value for that also. Let's see what the school providing us. See, the main purpose of showing this is that, see, everybody knows lake pH value won't increase or decrease in just one night. Okay, the main purpose of showing this is the model is.
Now it shows the value of 7.74.
Okay. Okay, my friends had doubt whether we can drink the Alva Institute of water. So I think I verified it now.
And your model is also tested.
Right. So to conclude, real-time monitoring is very important for the lake health. Okay, yeah, as I said before, the municipality waste, they should be treated properly before entering the water bodies. Okay, and we also yesterday Subhash Chandran sir, while he was giving his speech, he said a wonderful thing. He was like, put a fence around 1,000 acres of land and don't allow any human activities in it. In 5 years, you will have a beautiful forest which nature itself created. But when we try to relate it into our lakes, I don't think any human will leave the lake without touching it for almost 5 years. That's an impossible task. Okay, really impossible task. So real-time monitoring can very much help in this, so that the lake is not destroyed and we don't need to invest crores of rupees again restoring the lakes. Lakhs of rupees are better than crores. Okay, thank you. Okay, sorry guys. Actually, whatever the PHL, I said it before, right? We cannot connect it through USB, we have to connect it to a battery. And when we connect to a battery, nobody asks the question that where will we get the value? Why? Nobody asks the question, right? So this value will be displayed on an app called Blink IoT. IoT stands for Internet of Things, where we can easily transfer data like sensors or anything which will be used for robotics and all, etc. So this Blink IoT app, I have installed it in my tab, and I just wanted to, as you can see, I forgot to show it that time when pH was being tested. So as you can see over here, using this gauge, okay, it's ranges and the value is showing 6, maybe it's 6.9 or something, because I didn't wipe it after this one. So this will work. Let me just show you. So I'm adding it to a basic solution. I think it should change. Now it's here. Yeah, so it's changing over there. Okay, so this can be done through the Blink IoT app. Blink IoT app is free for using. That's why I installed this application to use it. There are many various apps which also supports many data types. And website is one more thing. And if given a chance, we can even develop apps. All these days, I think nobody goes to a website. We all use apps, right? Right? Yes. Okay. So that one thing can also be done. So these are the initiatives. Small steps can lead to a big impact in future. It can be positive or negative. Okay? Thank you so much. Thank you.
Efforts and time. Give him a round of applause. Thank you. I would like to thank ALVA's volunteers, NCC volunteers and teacher coordinator and also other volunteers for being with us from morning to evening and guiding us. It is time to felicitate and give all the certificates to all the presenters. Win or lose, first place or second place, you all have competed in a very tough race. Let me have a smile on your face. Now I request Aparna and sir and also Niyam sir, they will hand over the certificates. It is in order sequence. We'll announce and I request all the participants to come and collect your certificates. First is Nandini and team. Next, Sohail and Vignesh. Next, Sohail, Vignesh and Pavitra, please. Next, Shruti and Varshini. Give them a round of applause, children. Next, Diksha. Next is Diksha. Round of applause to Diksha. Next, Disha. Next, Lavanya. Himani. Next, Dharani. Next, Varshita and Minu, please come. Give them a round of applause for Dharani and Minu. Next, Maharashmi and Sneha. Applause to Kuti. Next, Tejas, Vihan and Atar. Next, Arun. Give him a round of applause for his continuous interaction. Amnish, Prasanna and Harini. They are not there. Next, Paulina. Okay, the same students. Niveda? Niveda and Amrutashri. Next, Mahi is Nityashri. Next, Shreya and Hema.
Shobhi is not here, no?
Yeah, Shobhi is not there. That's it.
And finally, Pratik.
Sir, we always join. Congratulations. Refreshment is provided. Coffee, tea. Coffee, tea is provided. put to right use. Correct? Data is like always use it or lose it. So let's use this. Young and energetic youth should come forward. I appreciate all the participants. Give a huge round of applause for yourself. And I also request sir to say a few words and motivate you all, sir.
Most of the things, competitions, people came here for the competitions. But when it comes to the presentation, I little bit wonder, because the student potential we never know. We should not underestimate, right? The people who present over here with a lot of ambitious, their struggle. Then the students who came over here to present calmly, nicely, right? Most of us, we observe that the younger minds are ignited more than the older ones, right? So that we are, but one thing is that they are supporting each other. We do appreciate they are supporting each other, they are clapping each other, right? And that is one of the very important. And one more very positive thing is that everybody answered with smiling. That is what we require in future. Right? See, today's media, we know that if you laugh, they will tag. If you smile, they will tag. And if you cry, they will tag. Right? So all these things, meanings, they will start creating the meanings. Like that, I did observe, even though many of us, I want to appreciate all the students, they never express any facial changes. That is what we need to require in future. Because today's media, your face expression changes, they will tag it. Right? So in future, wherever you go, you just cultivate this habit. Don't ever change your face expression in front of anybody. Whatever may be the situation. It may be joy moment, it may be happy moment, it may be, something, where you are loser. Then also you should not express any kind of facial expression. Because knowledge is there. Knowledge is there with all. We know that. Some people will have a little bit higher level, some people will have a lower level, some are getting that knowledge, right? So knowledge is there with all of you, utilize that one and continue the same thing in future. Thank you very much.
Thank you so much, sir. We always believe that students are 26 karat gold. No matter, teachers are very strong and bold. In spite of having fever and cold, we know when to hold and how to fold. Children, give a round of applause to all the teachers. Our teachers can shine. We all are here because of our teachers, correct? We always say, I always believe that teachers can, if things are fine, teachers can shine. And every teacher will treat all as like mine. Wish you all the best. We'll assemble in main VS Auditorium hall after your refreshment. Please do have refreshment. Use fresh wash, whatever, get freshen up, get some energy. We'll assemble in VS Auditorium hall for cultural extravaganza. Advas College students is known to perform a fantastic performance. You will not believe that students are performing. They are trained by professional artists, and today we are here and to explore and enjoy. We all will be wearing school uniform. Let's behave ourselves. We will maintain the dignity and we will enjoy. Mohan sir believes we should respect each and every artist only by clapping, not by shouting or cheering, only by claps.
I was known for cultural and sports, right? And for that about discipline. This is very much important. You may excel, you may excited during the performance, cultural performance. Kindly, politely clap it, but don't shout and don't visual it. I know that you are all young minds. You may over enthusiastically, you may do some things, right? Don't do that, because we maintain this decorum for long years. In every program, the performance, you can't believe it. Those are all students. Students are going to put their effort and they will perform better than the expertise. Better than the expertise. So therefore, kindly acknowledge their performance by clapping only. Thank you.
Yeah, tea is provided. Wish you all the best. Thank you so much. And round applause to all the IISC volunteers, NCC volunteers, teachers, and the wonderful hospitality and